Okay, hi everyone. So I'm Neil, founder and director of the Clinician Engineer Hub, and this is day 2 of our virtual conference. For those of you that attended day 1, I'm sure you remember all the fantastic speakers that we had across academia and industry, as well as healthcare. And it's always good to kick off with a speaker from the US for every conference. And we have again, by far, one of the most prolific speakers in the field of engineering, Professor Edward Boyden, direct from MIT. Professor Boyden, I'm sure you all are aware, has done massive amounts of work in engineering, particularly around the brain and psychiatry, looking at mapping, looking at novel therapies. So I'm sure all of you will be very impressed by what he's going to present. So Professor Boyden, over to you, sir.
Thank you. Really appreciate the kind introduction. Today I want to tell you about some tools that we've been developing for analyzing and repairing the brain and other complex systems. The brain is extremely complicated, and we don't really know how it works. We don't know how to treat most brain diseases. I switched from engineering into neuroscience 22 years ago, and in those 22 years, we have no cures and barely any new treatments for brain disease. So our group develops tools so we can understand the brain better and hopefully discover new ways to treat brain diseases. So today I'll tell you 3 short stories about tools that we're developing that are having impact on generating new strategies for the diagnosis and potentially treatment of different health-related conditions. Now, why do we develop new tools? Well, the brain has incredible spatial complexity and temporal complexity. Brain cells are high speed as far as biological systems go. And the wiring of the brain is very complicated. Brain cells are large objects, centimeters in spatial extent, but the wiring is nanoscale. So how do you see across these spatial scales, from nanoscale to macro scale? How do you control the high-speed dynamics of these systems? So I'll tell you 3 short stories today. 1 about space, how we make maps, and 2 about time, how we control and observe. Let's start with space. Lots of people have developed ways of nanoscale imaging, super resolution microscopes, for example, electron microscopes. But all these methods struggle to image large 3D objects, like brain circuits. So we invented a tool that we call expansion microscopy. This is now in use by many thousands of investigators around the world. How does expansion microscopy work? Well, basically, it's the opposite of regular microscopy. For hundreds of years, people have been zooming in with lenses to look at cells. That's how microscopes work. But we thought, let's do the opposite. Let's chemically create a dense mesh of polymer, anchor key biomolecules to the polymer, add water, and make the brain bigger. This technology owes a debt to some older strategies. In 1980, a paper from an MIT group actually explored the physics of swellable hydrogels. So these are highly charged polymers. You add water, the water is drawn in through osmosis, and the polymers swell. And the physics of this was worked out. Around the same time, 1981, Christine Dreyer and Peter Hausen had a paper where they figured out how to synthesize such hydrogels inside biological specimens. So they use chemistry to synthesize these meshes of polymer. They use an uncharged polymer, polyacrylamide. We wanted to use a charged polymer, polyacrylate. The charge is helping with the repulsion and making the thing bigger. So the basic idea then is, what if we can take a brain cell or tissue like the neuron on the left, and we can swell it to something like on the right, a constellation of biomolecules hovering in space with their relative organizations preserved. So these cartoons show how we do it. In this cartoon, the biomolecules are shown in brown, and we create handles shown in purple, little anchors that bind to the biomolecules. Then we weave that dense spider web-like mesh of polymer inside the cells, outside the cells, in between the biomolecules, around the biomolecules. You can see the little white spheres are the monomers, and they self-assemble the polymers, shown here. When the polymers encounter a handle, they form a bond. Finally, we soften the specimen using detergents, heat, or enzymes, or all three. We add water, and the polymer threads swell apart from each other. Because of the handles, though, the biomolecules come along for the ride. We call this expansion microscopy. In panel B is a piece of the mouse brain, and panel C is the same piece of mouse brain a day and a half later. We've swollen it by 100 times in volume. The polymer starts out very dense, like in the upper left, and it ends up extended, like in the lower left. The polymers are very, very dense. Their spacing is around the size of a single biomolecule. So in principle, this could preserve nanoscale information down to molecular scale. Here's a video. This is a piece of the mouse brain, which had a polymer synthesized earlier throughout, and we just added the water right there. I hope you can see the brain is swelling. The video is sped up, so it takes more like an hour than a minute, but I hope you can get a feel for how fast the process is. Is the expansion accurate? It's pretty accurate. It's not perfect, but the distortion created by the expansion is very small, maybe a few percent over length scales of tens to hundreds of micrometers, sort of the scale of a typical microscope field of view. And there's all sorts of things you can see with it. I'll just give you some examples here. By the way, this original paper was led by two then grad students in my group, Fei Chen and Paul Tilburg. This lets you do nanoimaging on regular microscopes. Also, because the expansion in water basically makes it 99% water because you're swelling with water, it lets you also use light sheet microscopes to scan even faster. Here in some work that Ray Gao, Sho Asano, and Gokul Padiyula did between my group and Eric Betzig's group, we expanded after labeling with fluorescent antibodies against mitochondria, lysosomal, and myelin proteins, as shown in these different panels. We expanded these fluorescent antibody-stained brain tissues and then used light sheet imaging to go extremely fast. You can also color code brain cells by expressing fluorescent proteins in them. If you deliver them randomly, then you get a combinatorial code shown at the bottom. This is called Brainbow and was developed by a group at Harvard. But when you zoom in on a microscope, you quickly hit the resolution limit shown in the top middle. In the upper right, though, you can see what happens if you expand in that image. The fuzzy structures in the top middle are now very clear in the upper right. You can resolve individual wires of the brain. Oswasi and Fei Chen worked together to implement a strategy for imaging RNA. In fact, here you can see some examples where they both anchored and expanded proteins shown in green and RNA shown in magenta. The net result is that you can visualize both of these with nanoscale precision on ordinary microscopes. How big can you expand? When J.B. Chang was a postdoc in the group, we worked on ways to expand brains twice. In the cartoon at the top, you can see how we do it. You can form a polymer shown in blue and expand the brain once, form a second polymer in the space opened up by the first expansion and expand it again. Recently, Deblina Sarkar, Oswasi, Jinyan Kang, with great help from Margaret Schroeder, we developed a way to do this using all off-the-shelf chemicals. Form a polymer mesh anchoring proteins to the polymer, form a second polymer in the space opened up by the first expansion, and simply stretch the first polymer further. This has the added advantage of decrowding proteins for better staining. Now we can look at very detailed structures like synapses shown at the bottom of the slide. The color code is shown in the letters above each panel. Importantly, by expanding proteins away from each other, we can decrowd them to visualize things you cannot ordinarily visualize. In panels B, C, and D, you can see us pre-labeling in yellow and post-labeling in magenta different proteins. As you can see, the magenta stains are much clearer than the yellow ones. That's because we're moving the proteins apart from each other and making room for the antibodies to bind. This does not hold for all proteins, though. Here at the bottom right, you can see that many proteins are perfectly fine with pre-expansion staining. Using this strategy, we can really tease apart the fine nanoscale architecture of synapses. Here's some work we did with Tom Blanpede's group, where we can look at very fine features of synapses called nanocolumns. We can see the distribution of proteins, presynaptic and postsynaptic, and see how they're organized. For example, here we can see how presynaptic calcium channels are coordinated with postsynaptic molecules like PSC95. Collaborating with Li Wei-Sai's group, we wanted to look at Alzheimer's disease. Amyloid plaques, for example, are very, very crowded. Here, again, in yellow, you can see pre-expansion staining, and in magenta, post-expansion staining. The post-expansion staining shows much more detail. In fact, you can see at the bottom, amyloid dots, these periodic structures that occur with a few hundred nanometer spacings that are not visible with pre-expansion staining. This worked with multiple antibodies and did not show up in wild-type mice. This is sort of a crowded slide, but it shows that we can analyze the detailed structure of these nanoclusters of amyloid. In fact, we were able to find within these clusters, subclusters of ion channels, which raises the question, could these amyloid dots have a role in the pathogenesis of Alzheimer's, perhaps contributing to hyperexcitability of the brain? Importantly, these strategies are not limited to the brain. We can apply them to all sorts of other tissue types. Here's some examples where we are expanding and looking at prostate, lung, breast, and pancreas. On the left, normal, and on the right, cancer-containing specimens from human patients. Lots of diseases are more treatable if you can detect them earlier. If you detect cancer earlier, for example, before it spreads, that makes it more likely to help the patient survive longer. Why is it so hard to detect diseases early? Well, it's often because the changes are so small, so subtle. We wondered, could we use expansion microscopy to make small changes more visible? Well, in this work, led by Yongxin Zhao and Octavia Bucure, we looked at breast cancer, collaborating with several pathologists at Harvard Medical School. Before expansion, we had a committee of pathologists grade these specimens of breast cancer from human patients. After expansion, we had the same committee regrade the specimens. Those votes were then used to train a machine learning algorithm. As you can see at the bottom, the machine learning algorithm was more consistent after expansion in green dots than before expansion, the purple squares. So we're very excited about this idea that perhaps we can use expansion to enhance our ability to detect diseases early. The expansion process is very simple to do. Almost 200 papers and preprints doing expansion have already come out. It's being used to explore the fruit fly brain, touch sensation, stress granules, embryogenesis, bacteria and the microbiome, kidney diseases, and the list goes on and on. Our website, expansionmicroscopy.org, describes how to do it. And appropriate to the post-COVID era, we also have been working on classroom-style, recipe-style strategies so anybody can learn how to do it. For example, here's a step-by-step tutorial that you can find on our website where you can take cells and expand them. It contains very detailed photographic documentation of each step. And hopefully this is enough to help anybody learn how to do this expansion process. So to summarize the first part of the talk, we've discovered that we can expand preserved, not living, biological specimens. This enables ordinary microscopes to become nanoimaging devices. This is being used to look at all sorts of biomolecules, such as proteins, nucleic acids, and other biomolecules. We can expand in one round about 4-fold, or in 2 rounds about 20-fold. And this is being used for all sorts of applications throughout biology and medicine. After all, biology and medicine are fundamentally nanoscale sciences. Gene products are nanoscale things. But up till now, it has not been a democratized process to do nanoimaging. But now anybody can do nanoimaging. I would estimate that over perhaps 1,000 groups are doing this kind of study. As I mentioned, almost 200 papers have already come out, and we've done hands-on training pre-COVID of about 400 groups, but hopefully the online tutorials are helpful for people to learn on their own. Now, one of the key limitations here is that expansion cannot be done on a living thing. So the next two short stories I will talk about are about how to control and observe the living brain. One strategy that we've been using to control the brain we call optogenetics. In optogenetics, we use light-driven molecules from the natural world to make brain cells activatable or silenceable with light. Then you can get light into the brain through optical fibers or other means. How do we do it? Well, we take light-activated molecules from the natural world and genetically express them in neurons. These molecules are called opsins. They're 7 transmembrane proteins that live in microbes and serve as a primitive photosynthetic strategy. They convert light to chemical energy. The first to be found was a light-driven proton pump about a half-century ago. Later, people found light-driven chloride pumps and light-driven ion channels. Flash forward to the present day, what we and many others are now finding is that these molecules can be found that are safe and effective in brain cells. From left to right, when Brian Chow and Shahan worked with me, we found that we could express light-driven proton pumps in neurons, use green or yellow light to activate these pumps, and turn the neurons off. In that way, you could figure out what the neurons are needed for, or you could try to shut down a pathological kind of activity. In the middle column, Xue and later Amy Chuang found light-driven chloride pumps that we could express in neurons, and these can be used to turn neurons off as well. On the right-hand side, light-driven ion channels can be found that allow us to turn neurons on with light. Carl Deisroth and I expressed these in neurons many years ago and found that we could activate neurons with blue light. And later, Nathan Klepetke, Alan Cho, or Shemesh, and others in our group have optimized these. What does optimize mean? Well, one thing that we've been doing is finding molecules that can be driven by red light. Red light can go deeper in the brain than other colors of light. Amy Chuang found a light-driven chloride pump that is red light-driven. Nathan Klepetke found a red light-activated ion channel. Both of these can be used to activate or control large volumes of the brain. The red light-driven ion channel is also being explored as a treatment for blindness. A group in France has just moved to the second stage of clinical trials. Patients with retinitis pigmentosa have lost their photoreceptor cells. But if you can restore to the eye light sensing, maybe people can see again. People are also using these tools to discover new patterns of brain activity that can be helpful. For example, Li Wei Sai's group used this strategy to drive the brain of Alzheimer's model mice at 40 hertz, 40 cycles per second. These mice got better. Now she's leading a strategy to try to drive these oscillations through sensory inputs, through flickering lights and clicking sounds. This is now being experimented on in clinical trials with actual Alzheimer's patients. In our group, we're trying to push the technology to its physical limits. Or Shemesh, Dimitri Tenesi, and Valeria Zampini, working between my group and Valentina Emiliani's group, have been working on optimized opsin microscope combinations. One idea is to take a molecule and express it at the cell body. Look at the left. If you shine a dotted circle of light at one cell, it'll hit the axons and dendrites from neighboring cells. So you aim the cell with the dotted circle, but you hit 3 cells with the 3 stars. In the middle, though, is an idea that other groups like McLean Bolton, Bajwa Pan, and Frank Werblins have explored. Can you aim the dotted circle at one cell and only hit that one cell? You can do that by focusing all the molecules to express only at the cell body. We did that. We found that we could take a molecule, a light-activated ion channel, and fuse it to a peptide so it only is at the cell body. Then when you aim light at those cells, they will be driven, but not their neighbors. So optogenetics, as this field has come to be known, is very powerful. It's being used by literally thousands of researchers to discover patterns of activity that can be used to initiate behavior, control behavior, or alter brain activity. Some of these are also having therapeutic insights. Now, the final short story I want to tell you about, though, is about a set of tools that we've been developing that fulfill another need. Can we see the brain in action? With optogenetics, we were very lucky. The natural world had evolved molecules that were fast enough, powerful enough, and safe enough that they could work in neurons. But what about the imaging of brain activity? In that case, we have not been so lucky. So when Erica Jung and Kirill Pyakovich were in the group, we decided, what if we could evolve molecules? If the natural world won't make these, maybe we can. We used a strategy that we invented, a robotic form of directed evolution. How does this work? Well, in directed evolution, you take a gene and make lots of mutants. Some are better and some are worse for any given goal. We transfect the mutants into cells, as shown in the top. Then, as in the bottom of the slide, we can scan those cells with a microscope and identify the cells that have the genes that we want. We then use a robotic arm to pull out those genes, to pull out those cells, and therefore those genes. With this strategy, we can make new molecules. The first thing we wanted to make was a fluorescent voltage indicator. We created 10 million mutants of genetically encoded voltage indicators based upon a molecule called Quasar-2, which was developed by Adam Cohen's group. We then used our cell-picking robot to pick out cells and therefore genes that had better brightness and localization. This is a crowded slide, but it shows that we were able to improve the molecule and make a new one that we called Archon. In the upper left, it was well localized to the membrane. In the top middle, it was brighter. In the upper right, it had better signal to noise. And in the bottom half of the slide, it had better kinetics and photostability. Now, using a microscope, like in the upper left, you can image the voltage of neurons. In collaborative work with Shua Han's group at BU, Kirill Pyakovich and Seth Ben-Sussan and Juan Tseng worked together to image neural voltage. What you can see here in the middle of the slide is neural activity that we are picking up on a camera. This is being imaged, not recorded with an electrode. Because it's picked up with a camera, you can image many neurons at once. Here you can see in an awake behaving mouse, 8 neurons being active at the same time. By the way, it's very important to focus the molecule expression at the cell body. Earlier, I told you how we do that for opsins, and here we're doing the same thing for voltage indicators. You can also do this for other indicators of neural activity. Here, calcium indicators are being expressed at the cell body. In panel A is a very popular calcium indicator, GCAM6. In B is the same indicator, but now expressed at the cell body. C and D are for a different calcium indicator. Why is that helpful? Well, nearby cells, if you image them, their activity can mix because light scatters in the brain. When you express the molecule just at the cell body, though, you greatly clean up the fluorescence. So we call this SOMA GCAM, a SOMA-expressed calcium indicator. This is work led by Or Shemesh, Chengyang Lingu, and Kirill Pyakovich in our group. Now, there are so many fluorescent indicators, hundreds at this point, that bioengineers have been making for decades. In the last story I want to tell you about today, I'll tell you how you can use these indicators together. Why is that important? Well, there's so many indicators, cyclic AMP indicators, glucose indicators, potassium indicators, and the list goes on and on. Wouldn't it be great to use them all together so you can map out a pathway in a cell? Well, Shannon Johnson and Chengyang Lingu in our group have figured out how to do this. The traditional way is you express 2 genetically encoded sensors in a cell, and then you have them be different colors. And then in your microscope, you can image the different colors at the same time. So if one indicator is green and one is red, then you can see both in the same cell. But there's only so many colors that you can see, right? Most groups can only see one or two things at a time. So we thought, what if we express different indicators at different points in a cell? Then each dot in a cell could broadcast a different kind of information to your microscope. How do you tell them apart, though? Well, later you can come back and stain the cell once the experiment's done and identify which indicator is at each point. We devised a couple strategies for doing this. Here's one of the ones that was the most successful. You can take fluorescent reporters, fuse them to self-assembling peptides, and create puncta inside the cells. Different puncta will have different reporters. If each reporter also has a different epitope, then later you can stain them with different antibodies, one after the other, and figure out which reporter was at each point. Here's GCAMP6, the calcium indicator I mentioned to you earlier in panel B. In panel C is what happens when we express the construct shown in panel A. GCAMP6 fused to two peptides. They self-assemble into little puncta. We did lots of experiments to test to see if this was safe and effective, and found no difference in the change in brightness, the signal to noise, the kinetics, and many other features, including whether any of these things changed as a function of the brightness or size of the puncta. Amazingly, this strategy was modular. We could take different glowing indicators and fuse them to different peptides and use them together in the same cell. So here we're taking three different indicators: GCAMP6, a calcium indicator, CAMP-R, a cyclic AMP indicator, and X-ray ACAR, a pKa indicator. These were made by other groups. We can fuse them to peptides and express them all in the same neuron. Panel B and C show what we get during the live imaging, the green dots, and after the experiment's done when we do the staining, shown in false color as yellow, blue, and purple. As you can see, we can identify in the living cell activity patterns by looking at the blinking of the green dots, and then when we're done, we can figure out what each signal meant. What that means is we can map out how these signals relate to each other. Here we're imaging three different signals in the same living cell. We can look at the relationships between these signals, which is important if we want to figure out how the cell works. In this case, we found patterns. Some cells had fast calcium responses, shown in I, and some had slower and choppier calcium responses, shown in H. These different classes of cell had different pKa responses later. This works in the living brain too. We can express these clusters in the living brain. And what we found was that in acute brain slices, the same categories appeared. Some neurons had fast calcium responses and some had slower ones. And those two classes of cell had different pKa responses. We can do this with 4 sensors at once. Here we're expressing 4 different indicators, all green, with different peptide tags that we then stain with antibodies afterwards. Then we can look at 4 signals at once, or we can even do 5 sensors. Here you can see 5 signals at once being imaged in a single living brain cell. So we can really start to tease apart the relationships between brain cells, which is so important. So to conclude the third short story I want to tell you about, with optogenetics, we were very lucky. With imaging brain activity, we have not been so lucky, but we can engineer our luck. We built robots that can evolve better fluorescent tools. We also have found ways like the soma targeting to make existing tools work better. And finally, we're figuring out how to use many of these tools at once to map out the patterns of activity in cells by clustering them at different points in space. I would like to combine all these tools to study entire small brains. If we could make a map of the brain of a small animal like a fish or a worm and see all its activity, maybe we could come up with computational models of brain function. This can be very powerful. So Limor Freifeld, when she was in our group, adapted expansion microscopy to the zebrafish brain. Jay Yu, when he was a PhD student in the group, did something similar for the worm C. elegans. And we've also adapted the voltage imaging to these small brains of fish and worms. So in the time to come, I hope that we can start to make computational models of brain function that could have broad utility in understanding how we can repair the brain. We distribute all of our tools as freely as possible. You can go to our website, synthneuro.org, or email me at edboyden@mit.edu. Many of these tools are in use by literally thousands of researchers around the world, and we're very excited to find ways to help people. So hopefully we can help you in your work as well. And with that, I'd be happy to take questions.
That was truly remarkable, sir. I mean, I think to achieve all that in such a short space of time is really, really quite incredible. I wanted to start off with a couple of questions, if I may. So obviously, as a clinician on this kind of interface between medicine and engineering, so my background isn't neurology, it's internal medicine and GI disease. But I have some experience in spectroscopy and microfluidics.
Ah, great.
I wanted to ask, this technique of expansion microscopy, has there ever been a situation where obviously during the technique, it can actually be problematic? So from a brain perspective, we worry as clinicians about brain edema and swelling, and that can obviously be a drawback. How do you get it to such a level that, you know, it's optimized so that it doesn't actually go the other way, if that makes sense?
So the expansion process, we only apply to preserved tissues. We don't apply it to living things.
Okay.
So when we work on the brain, we're getting brain specimens out of the operating room, or we're looking at Alzheimer's or Parkinson's brains from brain banks. So most of the applications are in more biomedical science than medicine.
Yeah.
The example I showed for breast cancer is more in fields where people routinely do take biopsies. So of course, in cancer biology and cancer medicine, biopsies are quite common. And so maybe I think the earliest clinical applications might be in early cancer detection.
Exactly.
But for example, you know, with COVID, a lot of questions are arising about whether the brain changes in COVID. And so we have a collaboration with Li Weisai's group to try to figure out, do these viruses get into the brain? And so we're looking at brain bank specimens now and trying to see if we can figure out where these nanoscale viruses are.
Great. And I wanted to ask, obviously your focus is on Alzheimer's, you mentioned. Do you think that there's a possibility for these techniques in, say, stroke or transient ischemic attacks? Because often, I think from experience, say we assume a patient may have had a stroke or a TIA, and we can image them, we do a CT, we don't really find anything. We may repeat the CT scan, or we may do an MRI, it may then show something. Do you think there's hope in the future that these techniques can actually look at the brain in more detail for stroke or maybe the TIA type patients?
It's a good question. I think it could definitely be helpful in understanding what's going on, which might lead to better ideas. I know some people have already started to try to apply the expansion technique to look at the blood-brain barrier and to look at other key features of the brain, trying to figure out how do these things work and how do they go wrong. So, but I think for the brain, it's quite possible that a lot of these techniques are going to be more on the biomedical science side than on the biomedical side. You know, studying the brain, just because of the inaccessibility of the brain to things like biopsies.
Perfect. And then I think just to finish off, because I know you're super busy, there was an element on your presentation where you spoke about the AI capabilities, looking at biopsy samples and that kind of thing. And we were having this discussion yesterday. Do you think artificial intelligence will take over pathologists in the future? You said that the outcome was that AI actually picked up signals better than a classic human observer.
Well, right now, most of the successes of AI are in doing things that humans do well. And I think that there definitely is a lot of excitement about that. I think, though, that for many of the diseases, especially for the brain, I think maybe you were kind of alluding to this a little bit earlier, we need to figure out more about what's wrong with the brain.
Absolutely.
You know, a given disease might have many subtypes. and being able to classify them might be very important. So I feel like there's still a lot of creative work that needs to be done in analyzing the types of brain diseases that occur, so we can be more targeted in our approach.
And I think what you say is absolutely right, because often, if someone comes in with a neurological presentation, half the time we're not able to piece it together. So the work that you're doing is looking at everything in more detail. So like you say, in the future, there may be new classifications and new neurological disorders that we haven't thought of. So that's really
Yeah,
fantastic. I think
yeah,
it's very, very exciting. Yeah, sorry,
right.
go ahead.
Yeah, these things are all connected. You need the science to drive the medicine and vice versa.
Absolutely. Well, thanks once again. I know it's very early for you. It's, I would have to say, definitely just an incredible amount of work that you've done in such a short space of time. So that's really absolute excellence. So thank you again, sir.
Hey,
Okay, everyone. So we're going to be back at 2 p.m. UK GMT for a talk from Microsoft. So we're going to take a quick break because it is a virtual conference. Stretch your legs, have something to eat and drink. We don't want you to sit down for the whole time. And we'll be back at 2 p.m.
Have a great day.
All the best, sir. Junaid, can you hear us? Perfect. Very nice background, sir. We definitely need that at this time.
I'm okay. Let me know when, but I'll.
Yes, okay. Absolutely. We're just preparing. Okay, we are ready. Okay, so hi everyone, we're back. This is day 2 of our Clinician Engineer Hub conference. We've just heard from Professor Edward Boyden talking in depth about all the advances in neurological disease. And we've had experts from around the world in academia, and now we've got experts from industry. So Dr. Bajwa is a practicing clinician. I don't know how he has the time to fulfill all his roles. Practicing clinician and chief scientist at Microsoft. And in terms of his background, he's looking exactly at all the issues in healthcare and how we can choose to solve these with technology. And I'm sure we're going to hear a lot of great things with him representing Microsoft. So Junaid, sir, we are ready when you are.
Okay, great. Thank you so much. I'll just share my screen.
Perfect. We can see it.
Amazing.
Great.
So hi everyone, and thank you very much for the warm introduction. So just as I mentioned, I'm Junaid, I'm Chief Medical Scientist at Microsoft Research. I'm a GP in Northwest London, and I'm a board member at UCLH. Feel free to connect on Twitter or LinkedIn if any of this is helpful information, and happy to catch up with delegates in the background as well. So I thought I'd maybe just start with a bit of my background and thinking through how I'm thinking about some of the challenges and problems, and bring this to life with some of the opportunities and experiences I've had. So the challenge that I've been set within Microsoft is, how do you begin to transform the practice of medicine with human-centered AI? And there's so much within that. First is, do we understand what the practice of medicine is today? Do we understand what the role is of medicine? How is medicine defined? And what is the role of AI? Over my past 15 years have been spent across the payer, provider, regulatory, and life science side of healthcare. So I trained as a surgeon, got my MRCS, I got hit by MTAS in 2007. I moved across into primary care, best decision I ever made, and started to see both inequalities and inequities in healthcare. And today I see incredible innovation Monday to Thursday. And when I'm in clinic on Friday, there's a massive delta between the innovation I hear about versus what actually is being delivered to my patients on the ground. And my job really is impact at scale and thinking about how I begin to reduce that delta. My role is global, so I get a chance to see what's happening across the world. And I'm deeply committed to the NHS and thinking about how we can bring that innovation to the NHS. And I'm constantly reminded by a quote from Peabody in 1926, which was, "The interest in humanity and the secret of caring for the patient is caring for the patient." Everything that we do, all this technology, should give us time back to care for patients and enable us to build on the superpowers that clinicians, physicians, nurses, and doctors have, and allow them to be better and more proficient at what they do. So if you think about how you begin to tackle some of these broad challenges and problems, this is from Professor Atun at Harvard. He would say, if you're thinking about analyzing what affects the adoption and diffusion of innovation, think about the context, think about the health system, think about the different actors, and think how ready that environment is to adopt that innovation. On a technology side, I think it's also about time. So what really can be done across the first horizon of time? What can be done across a second horizon? And what can be done across a third horizon? How ready is University Hospital Birmingham, for example, to adopt a Horizon 3 technology versus a Horizon 1 technology? And what is the role that clinicians, engineers, and others have to both establish and imagine a better future, but ensure that we're cementing on the foundations of what we need to solve for today? So in my mind, you can't imagine that better future unless you solve the challenges of today. And solving the challenges of today gives you the right and the permission to think about what you do moving forward. In terms of context, we also know that 5% of our populations across the world take up 20 to 25% of financial resources. These are those individuals with complex comorbidities, complex needs, often living with multiple different long-term conditions. So one of the first pieces of data analysis I did about 10 years ago was to look at my population in primary care. So this was a 5,000 list size practice, 80% of whom did not speak English as a first language. I wanted to do relief work in Southeast Asia. Someone said, do relief work in Plumstead before you think about Southeast Asia, where my population spoke Punjabi, Urdu, Hindi, Gujarati, Nepalese. I couldn't speak all of those languages, but I could empathize, and I could speak some of those to think about what their needs were. I began to realize that I was calling patients in for their chronic disease reviews, yet I had patients that lived on 13 or 14 different long-term condition registers. So I was calling these people in more than once a month. And then I was thinking about the difficulty I had for access. And when I was bringing them in, I was really thinking about a different question. I was thinking about what was the matter with them as opposed to what mattered to them. And as I started to change my conversation and narrative, I could empower them and think about what needed to be done. So just having that disease did not make them well or unwell. They could be perfectly well and perfectly managed if they were empowered to do so and felt safe in themselves. So thinking about the triple aim of healthcare, which I believe most clinicians aspire to and should aspire to, is how do we ensure that we're delivering population health at scale? How do we ensure we have good experience of delivering that care? How do we improve the experiences of care that we're delivering to our patients that we serve? And as good citizens, thinking about the NHS, how do we ultimately think about the per capita cost of spend on that individual? Globally, there's a series of factors which are shaping global health systems. Again, context. We're thinking about an aging population with increasing physical disability. As I mentioned, we have people not living with a single long-term condition, but multi-morbidity. People's expectations are rising. They almost want the Netflix on-demand version of healthcare. And we're about to enter a recession. And that recession will have an impact both on individuals' physical health and mental well-being as well. Yeah, we have a significant opportunity around innovation, leveraging technology, mobile devices, etc. We know by 2030, there'll be a shortfall of about 14 million healthcare workers, pharmacists, doctors, nurses across the planet, predominantly in low middle income countries. Yet their expectations will still be there and aging populations will still be there. So the challenge is, how can technology begin to innovate in the main and then around this? A lot of the work to date has been thinking about the medical sphere of data, but as we know, health is more than just your medical care. It's not just what exists within an electronic medical record. It is about the individual's behavior. It is about their social circumstances. It's their genomics, their economics, their socioeconomics, the environments they live in, to really think about healthcare. And we need to move the conversation, as I started to do 10 years ago, from understanding not just the disease state, but thinking about what truly matters to that individual and how can we start to focus on things that matter to them and empower them to live better lives and have high quality of life. Yet all of this data has massive jurisdictional constraints around it. So in different countries, there are both challenges with data, there are opportunities, and there are emerging issues and trends. So this is a chart from a couple of years ago, but in my mind, the key summary takeaway from this slide is, with great data comes great responsibility. So if we think about the challenges ahead, all of this data, we need to be responsible with it, we need to act in ethical manners, and we need to be very, very conscious around the regulatory environment that we live in, so that we're doing the right thing with the smallest amount of data that we need to make an impact on that individual's life. So just maybe taking a step back and synthesizing what we've discussed so far, we know that there are rising patient expectations. We know that healthcare systems around the world are transforming, and I believe over the last year, that transformation has catalyzed as a consequence of the pandemic. We know that the world will have less doctors, nurses, and pharmacists on the planet than what society will need. And we need to be conscious of their experiences and ensuring that they don't burn out. And we are equally conscious that there's massive fragmentation and silos in healthcare. And all of this is both an opportunity and a challenge. From a technology perspective, you've now seen over the last decade, increasing technology companies venturing into healthcare. The graph on the left here shows you, if you take from going from the printing press in the year 1400s to the innovation that we've seen post the Industrial Revolution, the technology industry has made such an impact in all of our lives. And organizations that did not exist 30 years ago are now making dents into healthcare, be that Microsoft, Amazon, Alphabet, Tencent, and others, with partnerships that they're forming, thinking about the opportunity to better understand the wealth of data that we have and how do we turn that data into meaningful insight that can translate with partners into better outcomes for the patients and public that we serve. From a Microsoft perspective, we're thinking deeply around the personalization of healthcare. How do we start to support with better diagnostics, to enable better therapeutics, to enable better outcomes? How do we ensure that we're enabling patients and individuals to understand their health, empowering teams, everything done in a confidential and secure manner to ultimately reimagine what healthcare could look like? So I thought, when speaking to Neil outside this meeting, I thought, how could I bring this to life a little bit? And I thought about the cancer journey, and thinking about not just from a traditional healthcare delivery perspective, but also from, say, where biopharma innovators, startups and others are going. And so there's a series of startups on the bottom who are trying to make an impact at different points across this linear description of a patient journey. We're hopeful that in our lifetime, cancer will become a long-term condition. But as it stands today, there are two ultimate outcomes from a cancer diagnosis. One is one of survivorship and the other one is of palliation. But the journey, and as we think about patient journeys as almost a lens into where we should focus all of our efforts, we must begin at the pre-symptomatic stage and go all the way to end of life. And what I've tried to articulate here is where there are both the challenges in the UK today and where the current pain points are. And why do we accept those current pain points to be true? And what could we do by leveraging technology or indeed analog solutions to improve how we do diagnosis, to improve how we do referrals, and improve how we think about treatment and care experiences, not just what happens when they enter and leave a hospital, but what happens in people's own homes. And we've seen announcements last week around what is happening with Grail in the NHS, and we'll see more announcements like this to come. Yet we know that the patient journey is not linear by any stretch of the imagination. Patients don't present and say, I have lung cancer. They present with a series of symptoms, and GPs, A&E doctors, generalists are doing a diagnostic sieve. And the picture on the left comes from the BMJ, who got together clinicians with designers to say, let's try and map out what this journey looks like. And the journey is complicated. And I wonder whether if we could better understand the data points within that, we could imagine a future, almost a Waze experience of healthcare, where there are choices to be made. Does the patient wish to go further afield to get treatment? Are they willing to tolerate certain side effects from their combination immuno-oncology drugs? Are they open to surgery or not open to surgery? What choices do they make to begin to personalize their cancer treatment and cancer care pathway? And the more we can do to think about earlier diagnosis, personalizing treatment options, what can we do to really think about what this means to personalized medicine? The knowledge is expanding exponentially. So every 30 seconds there's a new article published, and 4,000 new papers are published on PubMed every single day. Most clinicians can read maybe up to 10 a day max on top of everything else that they're doing. And medical knowledge is doubling every 73 days. So if I take cancer again, what is that choice of surgery, chemotherapy, radiotherapy, immunotherapy, and what combination of those is best suited for this patient, for their demographic, for their socioeconomic state, for the risk tolerability that they're willing to tolerate, around what treatment options are made around them at an NDT table, how that's communicated to them, and then how are they being cared for back home. So we will have an explosion of real world data at our disposal. And how do we make sense of all of this information and bring it to life? And this information is also in multiple different languages. It could be in French and German, in Mandarin, in Japanese. And so can we begin to leverage technology such as NLP, but make it intelligent enough to translate and then think about what that means for that moment in time diagnostic and care? And there's a lot of work on both NLP technologies that occurred during COVID in its early stages to think about how you democratize that information for the purposes of research. I imagine a world that hopefully, if you think about what Tesla has done for autonomous vehicles, and this is just a Tesla video on how the compute power takes a ton of features into consideration, could we imagine a better cancer journey than this, where we're anticipating the need for that individual well in advance and leveraging all the knowledge that is constantly being developed at our disposal to give that individual moments in time treatment options and moments in time better outcomes and moments in time better care? I thought I also might just show you a few examples of what has happened during COVID, leveraging things such as HoloLens. So Neil, with your permission, I have just 2 videos to show, and hopefully they'll work. If they don't, raise your hand, and then we can kind of move on to the next thing if it doesn't work. Should I give it a go? Yeah.
Yeah, go for it. Yeah.
Not supported. I'll put the videos onto the chat, and then you guys can watch them later.
Sure thing. Yeah, thanks very much.
This is a super interesting video. I'll just give you maybe the context for it. Oh, let's see. This one might actually work. Let's give it a go.
This one works.
You don't become a doctor to bill. You become a doctor to take care of patients. You become a doctor to serve humanity. Ambient intelligence allows for the patient and doctor to be a patient and a doctor, like it used to be.
There's a certain business requirement for a structured note and careful documentation, precise diagnoses, and carefully backed up coding for the business practice of medicine. But ultimately, it's about the patient's story.
If you look at the promise of meaningful use and all the reasons we got into a digital health record, it really was to take better care of the patient. Unfortunately, sometimes we've done that through a barrier, meaning we added a keyboard, we added a computer. I think that's a key part of why physicians are frustrated.
And I mean, you notice as a patient, you notice that your physician is typing and talking and, you know, yeah, okay, yeah.
Now we could go into the background and just capture all the information you needed, but also maybe more importantly, capture something that was missed. That missed was the patient's story.
There's a device on the wall, and it's listening to your conversation, and it's able to discriminate between a patient and a physician and a family member, and is able to generate a progress note and a patient encounter based on your conversation.
What it has enabled is that I can pay attention to what you are saying, because it is very important for me to have the empathy and you to feel as a patient that you are telling somebody who actually cares about you.
Hey, Dragon, start my standard order set for arthritis pain.
The idea of a computer that can listen like in Star Trek and just hear what's going on and make sense of it and slip into the background and listen, but only interject when necessary. That was for a long time a fantasy.
I would say before, in a typical week, I would probably spend 6 to 8 hours documenting, just simply dictating and signing notes. I bet that my time has been cut into half, if not thirds, because of nuance now.
It takes the hands-on time that the provider has to spend with the computer and puts that time back with the patient.
It really takes a lot of that manual work out of the patient encounter and just allows it to happen kind of naturally.
Ambient puts a listening device in the room quietly, securely, privately, and allows the 2 people in the room to really have a very intimate discussion on very private things. Knowing that the necessary recording of that for the business of medicine and healthcare can go on in the background, but letting that personal connection continue. And that's the reason I think almost all of us went into healthcare. It's to be able to be there for people, to express caring, to express empathy, and to connect as human beings.
So this is a partnership with Nuance, and this is live in hospitals also within the UK. And as a consequence of COVID, we've also powered that into Teams. So in Teams, as you're thinking about clinical consultations, there's now that automatic read coding that can happen in the background, published to OneNote, that you can then take out and extract into your EMR provider. And I believe that more of this will come in the future. Get back to the screen. Sorry, Neil, give me 1 sec.
Don't worry.
Don't become
Hopefully you guys can still see my screen. I thought it might be interesting for you just to know a little bit about Microsoft Research and our lab. So we have 8 labs across the world. all focused on actually a range of different things. So on the one side, we have this focus on medical health and genomics. But what's lovely is the opportunity for me to collaborate with people who are working on next generation Xbox, or the next generation of HoloLens, or people looking at finance and security, and then challenging me to think about privacy, confidentiality in different ways, and then taking learnings from other industries and bringing them together. So we all collaborate to then think about what that better future could ultimately look like. And so over the last couple of years, we've had a number of patents form, a number of publications and projects. You may have seen some recent news, and I'm happy to forward this on to Neil and the team around what we're doing for imaging. Some of this work is happening in Birmingham, some of this work is happening in UCLH around COVID, but really thinking about how do you open source some of our technology to allow clinicians and engineers to build upon it. So one of our technologies is Project InnerEye, which is thinking about the radiotherapy experience. And a publication that came out 2 days ago demonstrates that something that would have taken a few hours to do is now only taking a few minutes, and is truly empowering clinicians around what they need to do to give time back to patients. So with that, I will stop sharing my screen. Feel free to connect, and my thanks to colleagues and team members who've all been so kind to already send messages my way on Twitter and on LinkedIn, and I'll be sure to follow up.
Perfect. Thanks very much. That was really fantastic. I was just wondering if you have time for some questions, or do you have to shoot off somewhere else?
Yeah, no.
I know you're very busy. So I thought actually the way you presented this is very interesting. So there's definitely barriers to technology implementation. We were hearing yesterday from Helen Meach, who spent a lot of time knocking on the doors of various hospitals, and I'm sure important people, saying that the NHS needs more engineers, they need more people involved in technology. What are the kind of barriers that you faced, say, in the primary care setting? Because I think the mindset is that, okay, all this technology, maybe it just belongs in the hospital, and we kind of leave it there. Do you face any barriers from your primary care colleagues or primary care in general?
So I think people are enamored by the use of technology. They underestimate the struggle it might take to truly implement it into a workflow. So I think for all clinicians, physicians, healthcare workers, we train so hard to get to where we're at. And we're almost, our mindset is to operate in a certain way, and we have a workflow in our minds. So no matter what we do, if we're thinking about implementing or creating a change or a disruption, I think we immediately have to think about how does whatever innovation we bring augment the workflow today and make somebody's life easier. If we're not going to make somebody's life easier, how are we going to get through this? And also this thinking about perverse incentives, or indeed incentives that might enable those individuals to be motivated for that change. So I'll give you an example of a project from 2015, where the NHS had announced a national program of test beds that many of you may be familiar with. Simon Stevens, the chief executive of the NHS, came on stage and said, the NHS has more pilots than British Airways. We need to do something different. It can't just be about pilots. We need to think about technology, public-private sector partnerships at scale. And his office was sponsoring this. You had Sir Patrick Valence's predecessor, Sir Mark Walport, sponsoring this. They gave the permission. They were saying, what if we could create anywhere between 250,000 to 1 million patient-size populations for us to think about this innovation? And they wanted groups to go away and think about what needed to be done. The project that I led within that was focused on long-term condition management. It was fully aligned to the strategy. But actually, the challenge isn't what happens necessarily at the top. It's what happens when you're thinking about implementing this thing on the ground. Sure. So the project was, how do you predict not only the diseases that people have in primary care today, but could we begin to anticipate what diseases they'll have tomorrow? And if we could anticipate what they have, could we think about prevention in a different way and leverage telehealth solutions to manage, say, COPD, heart failure at home for a range of patient demographics and populations? This project needed us to then navigate 6 regulators, 3 payers, and 42 providers to do the right thing. And this is across a population in Manchester of 250,000 people. And it took a year of negotiation around the information governance aspect of what we believed to be applicable for us to do this work. And there were a range of actors and stakeholders. And the trick I think that we missed at the beginning was, do we truly understand what it takes to change. What does the university want? What do the GPs want? What does the LMC want, who represent the GPs? What does the hospital want? What do the community providers want? And do we begin to understand really everybody's needs, to think about the innovation and adoption environment for that innovation? And if we were to think about how we would do that again, I think deep stakeholder management and relationship building is almost as important as thinking through what the technology innovation might be. And if we can get that right up front, 98% is almost alignment, 2% ends up being the work. But if you create that environment, imagine what else you could innovate and build upon on that platform to truly, truly ensure that the North Star for everyone is improving patient outcomes and patient care. That has to be the North Star. And we have to constantly remind people of that.
Fantastic. So we do want to keep you busy for a bit longer, if that's all right. So we have our student representation as well. So we've got Chloe Jordan and Maria from Bartson London School of Medicine and Dentistry. We've also got Faisal, who's live from Dubai. They have lots of questions, I'm sure, and I'll hand it over to them to lead the discussion.
Thank you, Dr. Bajwa, for such an inspiring presentation. I have a question for you. Can you please talk us through the challenges faced with HoloLens throughout the COVID pandemic? Thank you.
So thanks, Maria. So I'd say actually it's more the opportunity with HoloLens. So I'll send across the video for the team to have a look at. But there's a surgeon at Imperial, his name is James Kinross, who decided that how is he going to be able to still think about ward rounds and at the same time have all the information available at his bedside at the peak of COVID. And so he started to use HoloLens to bring together the information at that moment in time and then use that at the bedside to then think about how that physician who could have been a junior doctor on their ward round could interface with the consultant who could be elsewhere. And at that moment in time, they were both collaborating with imaging, EMR data, and could see the patient together to think almost about what that virtual ward round could be. I can't remember if you're at Barts or if Chloe's at Barts, but you've also got Professor Shafi Ahmed at Barts, who's thinking about how does he leverage HoloLens for the purposes of education, knowing that actually some students can come in and others can't. And again, I think what I'm finding interesting is the surgical faculty that seems to be driving some of this innovation and transformation. Like with all things, I think you need to have those champions and sponsors who wish to think about the implementation in their respective organizations. And then there's always the challenge of how, if that works for them, how could it work for others, and thinking about a scale and a business case that might fit into the hospital. But I'll share the video with you if it's of interest, and feel free to share it amongst your community.
Thank you so much.
Perfect. Chloe, any questions from your side?
I think I've just seen in the chat that somebody's asked a question, so I'll just read it out. And it says, if you wait for all stakeholders to be on the same page, won't it delay the implementation of novel technology / interventions significantly?
It's a great point. So I think being aware of as many stakeholders as possible is super important. And chances are the first or second time you do this, you're going to miss some people out and you'll bear the scars on your back as a consequence of it. So at least mapping the stakeholders is really, really important. The more that you can align, the easier your work will become in the future. And I suppose it really depends on the nature of the innovation. So if we think about the COVID vaccine, that's an innovation. And we will do everything we can to make sure that it's implemented safely, effectively, and we're doing things at scale. And I think what we've almost seen in the last year is a wartime effort of groups coming together around a common understanding and a common problem, thinking about the implementation. What we also saw was as the peak died down, people reverted to old practices. They kind of went back into thinking about the added layers of bureaucracy that we could create to reduce some of that innovation. The question is, how do you maintain that positive mindset to get the right things done? And if you could do it for COVID, what could you do it for across other pathways? But it's not easy. And it's something, I suppose, I've learned it in the NHS. I've taken it into a pharmaceutical company, and I'm now taking that same mindset of stakeholder management and mapping into Microsoft. And it's a critical superpower that I would recommend anybody to experience and get for whatever career they wish to go into.
Thank you for that. And I guess one of the questions that's also come up quite a lot is, what advice do you have for medical students who are looking to go down the same path in the future?
So I suppose I have two rules. One is I wanted to make my mum happy, and so I still practice a day a week in the NHS, and I love it. And I didn't really have a plan, right? I've been opportunistic, and I've wanted to make a difference. My North Star, my job has fundamentally been the same. How do I deliver impact at scale? And I've pivoted, and I've taken a few risks. So my pivots were, when I was at medical school, it was all about becoming a surgeon. I thought that was the coolest job I could have. In 2007, I had to choose a different path based on both personal circumstances and professional circumstances. I then went into general practice, and I saw massive inequity in healthcare, which I felt that if I wanted to make an impact, I can do so much from a practice. What more could I do if I joined what's known as a CCG, so the payer community? But I didn't really understand that, so I ended up getting another qualification to make myself relevant, to come back, to have even broader impact. And then at every level you kind of see, just that, oh, okay, I've just done a different rotation. So my rotations in medical school, post-medical school, were I did A&E, I did general surgery, I did general medicine. I then moved into my CCG rotation. I understood what it means to be a payer, how local authorities work, how hospitals think about their budgets. I then moved into a pharma company and I did my rotation in pharma. And now I'm doing my rotation in Microsoft, thinking about big technology. But that rotational mindset that medics have, you could completely take into anything that you do. And life is a set of experiences. Always be relevant, always try and please your parents where you can. I think that will hold you in good stead. And just try and do the right thing and be a servant leader where you can and make a difference for those that you represent and serve.
No, that's some really, really great advice. And I think that's something that we can all take on board.
Fantastic. We have lots more questions for you, Junaid. So we've got Faisal from Dubai. Hopefully he can hear us. Faisal, can you hear us? We cannot hear you, Faisal.
Hi, yes, I think you
perfect.
can hear me now. Perfect. All right. Well, first of all, thank you, Dr. Junaid. I think it was an ecstatic presentation, to say the least. We really enjoyed it from the point of healthcare professionals, researchers, and young students who joined in for this conference. I think we're just honored to have such inspiration, keeping in mind that not all of us would be going into a traditionalistic path into medicine, but rather exploring a rotational mindset, be it in the health tech industry or any other field of expertise. So that was inspirational. We look forward to having more collaborations and talks like these in the future.
You're very kind. Thank you, Faisal. I appreciate the words.
Great. Any further questions from our student experts? Are they all questioned out? I have one question, if I may. So I think what you said is exactly right. It's how you kind of put forward the excitement about technology. So I was kind of mentioning this yesterday, that ultimately when I went through medical training, and I'm sure you're the same, this concept of taking a history, performing a physical exam, even though it's important, every step of the way when I'm on call in the hospital, I'm using some form of engineering solution to diagnose or manage patients. And then you have the kind of senior physicians, clinicians that are a bit anti-technology. But I think what you say is right. So that element where you're utilizing technology to look at patients in more of a holistic fashion. So recently, we have a massive inflammatory bowel disease cohort here. And instead of focusing on, okay, we must keep performing an endoscopy all the time, or we must keep imaging them, we're utilizing novel app-based technology to look at how they function on a day-to-day basis. So often what we ignore with chronic diseases is how are they functioning? What are their energy levels like? Are they able to form relationships? Are they having any issues at work? So do you think for the people that are resistant to technology, maybe that's more important to focus on that side, or what do you feel?
So it's interesting, right? So I'd say there's probably not a one size fits all, but I would take you back, Neil, to maybe that horizon piece, right? So why are they resistant to change? Have we understood their needs? Do they think, for example, that whatever this innovation and disruption is, that they're going to be out of a job? So that was the big thing for telehealth 10 years ago, right? It was, if we implement telehealth solutions, is there a group of people and when I say telehealth, I'm thinking about remote monitoring of blood pressure, remote monitoring of physiological signs and symptoms, not video conferencing or telemedicine. But telehealth, when they did a study on it 10 years ago, they added it to the current work. They didn't remove anything, and suddenly it became an additive feature. Yet the nurses who ended up having to take the burden of all of these measurements were saying, on a day to day, we might say 20 signals that we have to do something with. Now we're having to manage 70 signals, and this is increasing risk on us. We're just not going to do it. We're going to do it, we did it the old way. And so are we understanding whether we're introducing risk or whether we're taking anything away? Are we adding something to, or not? So I think there's a time component to these things that over time, like during COVID, it's been a complete blessing to be able to try and manage and monitor people at home in a very, very different way.
Absolutely.
Commercially, people talk about a product market fit. So is the product right for that moment in time for that market? And if you guys ever get a chance, there's a fantastic documentary called General Magic that I encourage all of your community to see. And it's the story of a version of a story of how the iPhone came to be. So it's the story about how the iPhone was actually invented in 1993 by a spin-out of Apple, who were a group of amazing engineers thinking about the next best thing after the Mac. This group of engineers ended up inventing the cloud, emojis, email, touch screen. What they didn't see coming, that was that the internet would be free. What they didn't see coming was that society probably wasn't ready for something that looked like this. And when they tried to sell it, even though they'd raised something like $60 million, they completely flopped and failed. But the individuals in that team, those individual engineers, one went on to essentially was the co-founder of eBay. The second one was the person who invented the Apple Watch. The third person invented every single generation of the iPod, and then invented how you take that iteration of iPod to the iPhone, because the iPhone wouldn't have happened if the iPod hadn't happened, and the iPod couldn't have scaled if it wasn't for iTunes. So it's this iteration of innovation that then led you to think through, actually, now it makes sense for me to use one of these things to do everything I want. And actually, how much innovation has happened as a consequence of this? So A, watch General Magic, B, product market fit, timing is everything. And then thinking about the unintended consequences of your innovation is an absolute, really.
Awesome, great. You seriously have the best job ever, very jealous. So thanks again for your time. If there's any more questions from Maria and Chloe or Faisal.
I actually think there's two questions that have just come through. So the first one was, in the case of technology use and digitalization, there are some critics about less human to human interaction. So how do we then balance this?
So I think where we are today and where we will be is what you are all being trained for, if you're clinicians, is how to measure up and manage risk. An algorithm's not going to do that today, and technology won't do that. Technology can give you additional information to help better understand the risk of the decision you're going to take. So I deliberately, as I think about the work that I'm doing, it's about the human in the loop, right? So how am I going to give more and more superpowers to the human in the loop? I don't think that we will be replacing humans in any given future right now. I think what we're thinking about and what we should reframe the conversation is, how do we give those individual superpowers and get them busy and operating at the top of their license in different ways?
Yeah, definitely. That's a great way.
Superpowers, that's what we need. So any more questions in the chat box there?
And we have one more question, and the question is, do you have any examples of healthcare technology innovation that led to an unanticipated adverse outcome? And what are the red flags to keep in mind when innovating?
Without naming any names, I think what we've seen is when people try and push things through and they haven't necessarily thought about the entire sequence of events, you can get a lot of noise, right? But the example I'm going to give you has also led, I believe, to some beneficial work. So there's an example of work that was being done at the Royal Free, and there was a question around how much the public were involved in the use of their data for the purposes of this particular innovation. That led to a range of questions around GDPR, appropriate use, And what that hospital was trying to do is really push the mold. And you could say that the innovation was super smart. It was to say, how do we get the right information to the right team member immediately? And that innovation did save people's lives. The way it was done and the way it was implemented could have been improved. And it led to a range of tabloid headlines. But there's been significant lessons learned from that, which organizations like NHSX, NHS Digital, and that hospital and others are now cautious, but they're truly thinking through the ethics of how that data is used and that personal information is used for public good. And those are very healthy conversations to have. I don't think we should rush these things, but we should be thinking through both research, translational research, and implementation of that innovation to think about how we do that seamlessly. And again, I come back to COVID. We've been able to solve a lot of these things during COVID. I wonder if it wasn't because of COVID, would we have had the permission to do certain things in the same way? And I see more and more research being translated, hopefully in real time, so that trials almost become a treatment option for many patients in the future.
Fantastic, brilliantly answered, sir. Okay, we will probably close there. Thanks again for your time. You know, it's an insane field, and I think, yeah, it's just going to keep growing. So thanks again. We're going to be back, everyone, at 3 p.m. UK GMT for our next speaker. So as I always say for these sitting down conferences, we should stretch our legs, have something to eat and drink, and then we'll see you again back at 3 p.m. Thanks, everyone.
1 second, I should be able to now.
Perfect. Okay. Oh, wow. Very nice background.
Oh, thank you. It's one of our Mayo virtual backgrounds. This is outside the chapel in St. Mary's Hospital, one of our major hospitals.
Amazing. Okay, we'll just wait a few more moments.
Yeah. So, how's the audio? You
Yeah,
through
audio's
okay?
perfect. We can see you fine. I'll introduce you, and then we'll be ready to go. Okay.
All right, and I'll just get my presentation up here so that I am ready.
Sure. Okay, well, hi everyone. This is back again for day 2 of our Clinician Engineer Hub virtual conference. We started off in the United States, came back to the UK, and now we're back in the US. And we have Mark here, who's the head of engineering, developments, and innovation at the Mayo Clinic, which is really incredible, you know, to have you spend the time with us today, because I'm sure you're very busy. So Mark has an insane amount of experience in industry and engineering. He's been doing this a very long time, much longer than I have. So it's very great to have his expertise here, and we're looking forward to your presentation, Mark.
Thank you, and I certainly appreciate the opportunity. Make sure my screen share works here. So,
yeah, we can see it. Perfect.
All right, excellent. Okay. Well, again, Neil, thanks for the introduction and the opportunity to speak to folks today. I gave a talk to the Clinician Engineer Hub last summer, and I'm really pleased to have another chance to do so. I've really spent my whole career partnering with clinicians and researchers with the idea of creating something bigger than what we could do individually. And I'm going to talk a little bit about that partnership today. And I think despite the mathematical dubiousness of the claim, I think there's times when 1 and 1 really is more than 2. A little bit about the work done by engineering at Mayo Clinic, and then I'm going to talk about a specific and pertinent example of engineers and clinicians working together. Know that we've got big challenges around the world with getting people to wear masks. In fact, last spring, we weren't even sure if they would help at all with the coronavirus. So I'm going to talk a little bit about how we partnered with our physicians to bring a little bit of clarity to that particular issue. But before I get started, let me do just a quick introduction of myself. So my name is Mark Wehde. I've got a bachelor's degree in electrical engineering, a master's in biomedical engineering, and an MBA. Started at Mayo right out of graduate school, and I spent the first part of my career developing medical devices, focusing on sensors, electrodes, analog circuit, microprocessor design. And I've also done lots of programming, embedded systems, PCs, mini computers. But really for the last 20 years, I've been a manager. I've managed our electronics and software development groups. I managed our mechanical unit and our machine shop. I even started our first project management office at Mayo back in about 2000. And most recently, I've taken over as chair of the Division of Engineering. Mayo is an academic healthcare center. This means education is a big part of what we do. If you look at the Mayo logo, we have 3 shields, and our 3 shields are research, education, and clinical practice. So that means one of the fun things I get to do is teach. So I'm an assistant professor in our biomedical engineering PhD program. I teach in our quality academy and our Office of Leadership and Organizational Development. And I also teach at the University of Wisconsin as part of their online So Mayo Clinic is the first, largest, and many would say the best integrated not-for-profit medical group practice in the world. And some say that this is actually the Mayo brothers' greatest contribution to medicine, was this concept of a medical group practice. My group, the engineering group, grew out of the rapid proliferation of surgical tools that occurred after the development of anesthesia and aseptic surgery in the late 1800s. So early on, the Mayo brothers and their partners would create their own surgical tools, but they recognized eventually they needed a dedicated group of technicians and machinists to support the growing complexity of the tools. And so you see an early picture of our machine shop. Early shop was formed in 1915, so 105 years ago. And here you see an article describing the manufacturing of screws using beef bones and a lathe in the machine shop. And these bone screws were used in the repair of femur fractures, and we made similar devices for spine stabilization. But in 1948, that group was reformed as the Division of Engineering. So we brought in engineering staff to supplement the technicians and machinists and glass blowers. One of the first things we worked on was what would become the Mayo Gibbon heart-lung machine. This was the first commercial heart-lung bypass machine, and it was first used in 1955. And according to the Smithsonian, the invention of the heart-lung machine is one of the most significant contributions in the history of cardiac surgery. We also helped with the development of the G-suit in the 1940s. This was a top secret project at Mayo that was part of our World War II effort. And early patient monitoring was developed by engineering. Nowadays, we're all familiar with all of the established commercial companies that are producing patient monitoring equipment. But in the early days, they didn't exist. And the engineering group at Mayo was developing our own patient monitoring equipment. But it was the invention of the transistor in the 40s, integrated circuit in the 1950s, the introduction of microcomputers or mini computers in the 60s, and then the release of the first commercial microprocessor in the early 70s that have allowed us to really create the incredible medical instrumentation that's used today in healthcare. And also in the late 70s and early 80s, we assisted with the development and research on the first high-speed CT scanner, and this was installed at Mayo in 1975. So our group, the engineering group at Mayo, our mission is to build unique medical devices to enhance patient care and advance medical research at Mayo. We haven't been asked to save Mayo money by making things cheaper, and we haven't been asked to make something that someone else already makes. We've been asked to help create new devices to better serve our patients. We're kind of a unique resource. I'm not aware of any other academic healthcare center that has an applied engineering group like ours. We're an integral part of the organization, and together with our physicians, we work at that intersection of engineering and medicine in ways that would be difficult if we weren't an integrated, embedded part of the practice. So some of our work includes some early telemedicine systems. We do a lot of computational. We do various surgical instruments. This is a device to allow the repair of the mitral valve without repair the chordae on the mitral valve while the heart is still beating. Emergency designs like these clips that were added to a commercial dialysis machine in response to a sentinel event. We've worked on a prototype bioartificial liver, which has sustained life in animal trials for 72 hours, and a variety of neurological devices. And I'm going to talk a little bit more about those in the next couple slides. So a major effort of ours over the last 10 years has been this collaboration with the Neural Engineering Lab to create a closed loop deep brain stimulation system. So deep brain stimulation is already an established treatment for a variety of mostly tremor disorders. But the goal of this program is to create a closed loop system to treat disorders such as depression, which don't have objective measures of the effectiveness of the treatment. And we're doing this through the development of a closed loop deep brain stimulation system that controls the stimulation through the measurement of the neurotransmitters that are released subsequent to the stimulation. So part of that is developing the hardware that lets us explore this new modality. And this includes a prototype animal use device. And we're now working on a third generation of the hardware in support of Institutional Review Board approved studies on humans. So we'll be using this to develop the closed loop feedback algorithms and also to test new versions of the neurochemical sensing electrodes. And that's the other piece of this project is the development of those electrodes. They're developed in a custom diamond reactor that we've created in our engineering labs. This uses a hot filament chemical vapor deposition reactor with an infusion of methane, hydrogen, and trimethyl borane to create boron-doped conductive diamonds. And in this view, you can see what the electrode actually looks like. So there is let me turn on my laser pointer here. Sorry. There we go. So this is the conductive diamond tip right here. And this is an insulating layer of parylene. And so this is the electrode that we're using to sense neurosensor or neurochemicals. And just to give you an idea of how this is working, let me see if I can get this video to play. There we go. So the electrode is inside this rat, and what you can see is the level of dopamine. And then we introduce another rat, and the dopamine level starts going down. Now, in a moment, the rats will get close enough that they recognize each other, and there you can see that spike that occurs when they got close together. So this is just a really good indication of how well those neurosensing electrodes that we've developed work. So we've also done some other work. We're working with our epilepsy teams as well to try to eliminate the need for a craniotomy when they're identifying the focal point of the seizure. We've got 2 significant efforts underway, including the development of thin film polyamide-based electrodes. So we have a variety of equipment that allows us to fabricate these electrodes in-house. We're also working on the deployment system. So our goal is to replace this very invasive craniotomy with a small burr hole through which we can put in an expandable electrode array. The device you see on the bottom right is right here is a 3D printed prototype of the device for inserting the electrodes that is being used here on cadavers. And now we have a new design that's made of titanium that we're using to evaluate this. And here's an example of the electrodes that we're making in our lab. So these are a combination of macro and micro electrodes that allow us to selectively cover larger and smaller areas of the brain. This lets us explore how the seizures start and end, and hopefully find better ways to stop them. And when we were working on that device that allows us to put the electrodes between the dura and the brain, our neurosurgeons had this idea of a modification which could be used for subdural hematomas. And so I'm going to give you just a quick video clip showing how that works. And this is actually another device that we have developed and have taken into clinical trials. So we got a mechanism that we attach through the burr hole, and then the catheter is guided into the subdural hematoma and then used to very carefully drain the fluid out of it. And this is an example of one of those things that just happens, I would say in some cases by serendipity, because what we were working on was how to put electrodes onto the brain, and the neurosurgeon said, I've got another idea. And so with some very simple modifications to that same mechanism that we used to slide the electrodes in, we were able to slide the catheter in that he used to drain the hematoma. So we can also now create novel surgical tools, and we can 3D print them right at the point of care. And we can also create sophisticated simulation systems that we can use for training and practice of surgery. So what you see on the screen is a model of the descending aorta. This is created from this one would be created from MR images of aortic aneurysms, and it's used to practice the deployment of stents for repairing the aneurysm. So this is used in a surgical suite to practice the surgery as realistically as possible. So in this conceptualization of it, you can see the pump for pulsal blood flow over on the right-hand side of the screen. And then on the left side, you can see the fluoroscopy system. So the surgeons are going to do surgery on this model just like it was a patient. And this is incredibly valuable as a training aid for new surgeons and to practice complex procedures, even for experienced surgeons. You can see a quick look here of how this thing works.
We actually have to do all the steps of the operation as they are, and it anticipates difficulties, challenges. Sometimes it helps answer questions. Connect this to a pump of fluid that has normal temperature that we have, about 35 to 36 degrees Celsius, and blood pressure.
So you get a sense of how we use that in our practice, and it's been really a huge boon for the surgeons, in terms of training, but also for practicing really complex procedures. We also do a lot of work with additive manufacturing. So we actually do both additive and subtractive manufacturing. We've got several 3D printers. We've also got a full machine shop. My introduction to 3D printing goes back to 2006. Mayo was doing surgery on a set of conjoined twins. So Abigail and Isabel Carlson were successfully separated by a team of Mayo physicians when they were 6 months old. Surgery on conjoined twins can take months to prepare. So in this surgery, there were 17 surgeons and over 60 staff participating in what ended up being a 12-hour surgery. Dr. Moyer, the lead surgeon on the left, asked us if we could help provide 3D printed models of the organs to be separated. So these girls were joined at the chest. Isabella's heart was in Abby's chest and their organs were twisted together. And Dr. Moyer was looking for models to allow him to look at the internal structure of the organs. So you can see in the upper right, the rotating image shows the liver complete with bile ducts and blood vessels. And then we printed that in a translucent model that he was able to take with him to planning sessions for the surgeries. And here are Abby and Belle today. They're happy and well-adjusted teenagers, and they come back for visits to Mayo every year. And you know, when we think about it, this is the reason that people go into medicine in the first place is for these kind of results. And for engineers, it's one of the reasons that working in this field is so incredibly rewarding, because it's one of those engineering fields where you can really see the impact of your work on the patients. And at Mayo, we talk about the needs of the patient come first, and we really do believe that, and all of us have the opportunity to contribute to it. Well, after the twins, we started making a whole host of different anatomical models for complex surgery. And these models will be used by surgeons for planning, practice, and also as guides for their surgery. And then in about 2013, we created a dedicated anatomical modeling lab as part of radiology. And so now that group can do these beautiful anatomical models with their current generation of printers. And they're now making over 3,000 models a year to help guide some of these very, very complex surgeries. So I'm going to share just a little bit about how we use 3D printing to help with some of these surgeries. In this particular example, we start with MRI images. Patient had a tumor in the lower mandible, and that part of the bone was going to have to be removed. So the part down here. And so we create a 3D model of the patient, and then we're able to visualize the mandible and the tumor to be removed. And we can add cutting guides. So these are the cutting planes for the surgeon to remove. And you can see from the side, so we can make sure that we're able to clearly identify the boundaries of the tumor so that we know that it's all going to be removed. And then we can virtually remove the tumor and the surrounding bone. Now we have to replace it. Well, so first of all, I guess to do those precise cuts, we create these custom cutting guides from the anatomical models that we have. So this will attach to the jaw with small screws, and then the surgeon can use it to cut precisely the right location to remove that bone containing the tumor. And then we have to start looking at replacing the piece that we pulled out. So this is what that replacement jaw would look like. Now at Mayo, we use a procedure called a fibula free flap. So this is a technique that's commonly used for mandible reconstruction. So you take a long bone, generally the fibula, and harvest that and cut it and mold it to the proper shape to replace the jaw. And so in the upper left, you can see how that fits together. So using this model, we create the custom guides to also create the new jaw from the patient's own fibula. So the original fibula is shown on the top, and on the bottom, you can see the cutting guide mounted to the fibula, and the surgeon will use these to make the appropriate cuts. And then when it's finished, this is how it goes together and what it'll look like. And we also need to provide fixation for the new jaw. So this attachment piece can be a flexible piece of metal that you bend in shape right during the surgery, or even better is to prepare it in advance. And the way we do that right now is by printing a model of the jaw and fitting the supporting structure to that model before we take it into the surgical suite. What we're going to be doing in the future is we're actually going to be 3D printing this on a metal printer, and that's going to be used to support the implanted jaw until it heals. Part of our work also is helping orthopedic surgeons with new designs for orthopedic implants. So this is a stemless humeral implant and a fracture stem implant. These were designed, 3D printed, and tested in cadavers. And then we also printed 3D metal versions so that we could do full strength testing in our biomechanics lab. So this is the problem we were trying to resolve. The commercial device simply didn't allow us to match the inclination to the anatomy very well. And the orthopedic surgeon wanted to create a design that allowed for more adjustment. So if we look at again, the original design, here was the model we came up with, which added additional degrees of freedom, and then the 3D printed polymer plastic model that we created to test this. Spinal fusion is another place that we've used 3D printing. So spinal fusion is a common treatment for facet and disc disorders, and they typically use titanium rods to maintain a fixed spacing between vertebrae. But this rigidity really limits the movement. So one of our neurospine surgeons had an idea for developing a novel device to allow the spine to move while still providing the required stabilization. So we start with these computational models, and we can do virtual surgeries with them, and we can design a variety of different implants. We can put them in, we can test them, we can measure some of their properties. So we start with those computational models, and then we can move from virtual to physical models for testing and evaluation. So we start with the computational model. We then make physical models that we can use with cadaver testing. And here you can see a picture of our prototype device. So Mayo Engineering is planning our next adventure in 3D printing with a titanium printer, and we're hoping to be able to print custom implants for animal trials and human clinical trials within a couple of years. There have been huge advances in 3D metal printing in the last 2 or 3 years, and that's really made the printing of custom implants at the point of care a very viable possibility. So we're working with the Food and Drug Administration here in the US to address some of the regulatory challenges, and we're really poised to be on the forefront of moving personalized medicine into custom implants. And the global 3D printing medical device market is going to be generating over $2 billion in revenue by the end of 2024. Manufacturers use these printers right now to develop inexpensive models like spine cages or cranial implants. We can also use them to create patient-specific implants using CT or MRI scans for complicated surgical procedures. And this point-of-care 3D printing is also a great training vehicle for 3D visualization for physicians and for manufacturing of non-implantable medical devices like surgical models or the cutting guides we talked about a little bit earlier, or in this particular model as supports for traditional implants. So currently, 3D printing of patient-specific implants is expensive and it's time-consuming, and this can take weeks or months to develop. But by putting this additive manufacturing right at the point of care, we think we're going to save time and money, and we're going to drive innovation because we're going to have the technologists and the engineers and the clinicians all working in this collaborative environment to come up with the best way of doing this and the best implants for the patients. Now, a metal printer isn't the only way to print point-of-care devices. One of the cool new applications of neuroprostheses and 3D printing is the creation of prosthetic arms. And this is a quick video from a company called Open Bionics. It's called the Hero Arm.
Open Bionics is a Bristol-based startup, and our mission is to create and democratize technology that enhances the human body. So in other words, we turn disabilities into superpowers.
So I think this bionic arm is one of the coolest ideas of what we can do with 3D printing, because it's really a great equalizer. It no longer takes millions of dollars to create a prosthetic arm. That particular arm in that video, the Hero arm, costs about $700 to make. So I think that there's just some huge possibilities for taking that technology around the world. Before I leave 3D printing, I see there's a question in the chat window I'm gonna address. So there was a question of, what files are used for 3D printing in a hospital setting and how easy it is to get those files. So the files come off of a CT or MRI in a standard format. It's an IGES format, and there are software programs out there that quite easily convert that into a format that we can use in SolidWorks, Materialize, Mimics. There's a variety of software tools that we use to do this, but it's very straightforward. Companies have been doing this for 20 years, and so the conversion from the format of the CT, MR, X-ray images, all of that it's a very straightforward process to go through. The hard part is the segmentation. So if you think about having a CT image, you've got a lot more things there than what you want. If you want an image of the fibula, it's also surrounded by muscle and blood vessels and things like that. And so the process of segmentation is what you go through to remove all of those things that you don't want. And that takes some real expertise. That's where our clinical partners come in. We have to have them help us define those boundaries between different types of tissues. That's simply not something that an engineer is trained to be able to do. And that's where this partnership and being able to sit together and look at the model is really, really helpful. So I'm gonna finish here with some work that really demonstrates the partnership between clinicians and engineers. And it's interesting to me that the study of masking and the use of masks has become a political fireball in the United States and elsewhere, where it really should simply be a scientific question. So my focus in this project was answering a very simple question: do masks work? Government leaders can decide if we use them. Engineering and science can only tell us if they work, and then that information can be used to guide policy. So with the coronavirus and COVID-19, it's thought that the primary means of transmission is through exposure to respiratory droplets that are formed when the person who has COVID-19 coughs, sneezes, sings, talks, or breathes. There's also some growing evidence that smaller particles called aerosols can remain suspended in the air and be breathed in by others and travel distances beyond 6 feet. So for example, some of the anecdotal evidence during choir practice in restaurants and fitness classes. In general, having an indoor environment without good ventilation is going to increase the risk of aerosol transmission. Now, aerosols are typically particles less than 5 microns in diameter. They are so light that gravity has very little impact on them. They can remain suspended in the air for hours, and to remove them requires either outside air exchange or filtration. In Mayo, in our test labs, but also in many of our procedure rooms, we've used standalone HEPA filters when we need extra clearance. We've also upgraded our HVAC systems when we can to a standard called MERV 13, which is an HVAC filter which can remove viral particles. Droplets, which are thought to be the primary mode of transmission, are particles generally considered to be greater than 5 microns. So these droplets are impacted by gravity, and they're readily filtered by conventional cloth and disposable masks. If we think about filtering mechanisms, there's 5 basic mechanisms. Gravity sedimentation means that the particle is heavy enough that gravity pulls it to the ground. In this case, we might end up with particles on surfaces, and that's also a problem to be aware of. But that is where this guidance for 6 feet or 2 meters has come from, is because these particles are generally pulled to ground relatively quickly. Inertial impaction. So that simply means a large particle runs into the filter material and is stopped or redirected. And this is the primary mechanism for filtering large respiratory droplets. Interception occurs when a smaller particle makes contact with a fiber in the filter and is captured. And this mechanism of action is a little bit different because the smaller particles have a lot less inertia than the bigger one. But this is really an effective mechanism for the filtration of smaller respiratory droplets. Then diffusion is really a mechanism for the extremely small particle. Those would be less than 0.3 microns. So a coronavirus particle on its own, without being in a respiratory droplet is about 0.2 microns. So this mechanism, diffusion, is due to the deposition onto the filter material due to a random Brownian motion. And this is an effective mechanism for small viral particles, again, that aren't part of a larger droplet. Aerosolizing procedures are known to produce these very small particles, and that's why N95s are required for those kind of procedures. And then finally, electrostatic attraction. This is based on the attraction due to different polarities of charge on the particles in the filter materials. And this is also a primary means of filtration for ultra small viral particles in N95 and HEPA filters. This graph gives us a sense of how far particles can travel versus size, and it's on a log scale to make it a little bit easier to see, because there's some dramatic changes with distance, which I'll show you in a minute. While this is somewhat dependent on the initial velocity, the different curves here represent different velocities. It's primarily dependent on the size of the particle. So that's because the inertial effect is the primary transport mechanism for larger particles until they start to lose some of that initial velocity. So if we look at that on a linear scale, you can see it even more dramatically. So you can see here out at past 10 microns, this particles simply don't travel very far, or less than that. They don't travel very far through the air via inertia. They simply float along in the airflow, whereas these larger particles have enough inertia that they move in a straight line until friction reduces their velocity. This is a graph of filter efficiency versus particle size. So most filters are least efficient right around 300 nanometers or 0.3 microns. So N95s are 95% effective at 300 nanometers and much more effective as we get to larger and smaller particles. And HEPA filters are even more effective than the N95s. For the larger particles, the primary mechanism for filtration then is impaction and interception. And that's why the cloth and disposable masks work so well for larger respiratory droplets. So the filtration of cloth and disposable masks is most effective for particles with significant inertia. And that's why the source mask is so much more effective than the target mask. So air comes from your lungs at a relatively high rate of speed, and the further it travels, the slower it goes. And by the time it's traveled a meter or two, it's lost most of its inertia. So the filtration mechanisms of impaction and interception don't work very well on the target mask. But you know, the cool part is that all masks collect these larger droplets during exhalation. And if we look at the collection efficiency of mass versus particle size, the first thing you see is that N95s are pretty linear all the way, you know, for all particles, and that's pretty nice. But when we look at smaller particles, surgical mask, cloth mask, and especially poorly fitting cloth masks really don't do much for us. It's only when we start getting into this larger range between 1 and 10 microns that we start seeing cloth masks starting to be effective. And then beyond 10 microns or so, the cloth masks are very effective, approaching that of N95s. And as you can expect, along the bottom here is a mask with an exhalation valve, and those simply don't filter anything. And of course, that's the problem with those type of masks, like the N95 with an exhalation port. You simply don't filter anything that you're breathing out. So here's what masks do. So masks will efficiently collect the larger particles and droplets from coughing, sneezing, and things like this, even if they're leaky. So this is what we engineers call source control. We're controlling the source of the virus. They also slow down the speed at which those particles move. And so this is what we call an engineering control. So we've designed a system that slows those particles down. And what they do is they make social or physical distancing more effective. And they also make ventilation more effective because they're reducing the number of particles that end up in the air, because many of these large respiratory droplets that you breathe out will evaporate as they travel through space and turn into aerosols and will be, can be floating in the airspace for a substantial amount of time. What they don't do, none of these masks provide 100% protection. They don't replace the need for physical distancing. They supplement it. They make it more effective. And they definitely don't eliminate the need for good ventilation. So when you have people inside, in an inside environment, even if you maintain distance, even if you've got masks, if you're going to be in there for a long period of time, you really need to have good ventilation. Let me give you now an idea of how we went about testing this. So we created an experimental setup based on a custom breathing simulator to nebulize water and mix it with 2-micron polystyrene beads, which was then pumped from a mannequin's mouth to simulate breathing. And we used those 2-micron polystyrene beads because we wanted a fixed size. And if you recall from the earlier slides, that's just in the range where masks, cloth, and disposable masks start to be effective. It wouldn't have been much use for us to do a study with 20 or 30 micron particles because there's simply no question about whether it's effective on those. We wanted to go back at this transition point where masks start to work. So this is the setup that we use. So this was the nebulizer. This is where we pumped in the 2-micron polystyrene beads and created the airflow, and then that is attached to the mannequin's mouth. Here's this kind of a size comparison. So the polystyrene particles are 2 microns. An individual coronavirus is 0.2. So these respiratory droplets, these larger droplets are from an infected person are very likely to contain many coronavirus particles contained within each of these droplets. So they simulate those respiratory droplets then. So we also used a respiratory mannequin as a target. So we have a source and we have a target. And on the target side, we actually pulled air from the nose and the mouth both and ran them into an aerodynamic particle sizer to count the particles that made it not just to the second mannequin, but were also breathed in by the inspiratory mechanism. And we used a design and experience methodology. We created a comprehensive battery of over 500 independent tests running at different distances, different directions, different type of masks to make sure we fully understood the movement of these respiratory droplets. We also had to use HEPA filtration in the room because we had to reduce the level of background particle counts in the room between experiments, because otherwise the background particle count overwhelms the particles that we were trying to measure. Now I'm going to give you just a quick here's an image that shows us. Now this is what is called a smoke test. It's not the experimental setup, but what you see is the difference when you have a mask on versus when you don't have a mask. Smoke is actually a good analog to use to try to visualize this. This is not how we did the test because we used that stream of 2-micron particles because we needed to make precise counts. But smoke is very similar to what you breathe out. It's got particles of many different sizes. So when you look at that video, let's see if I can play it again. There we go. So you can see without the mask, inertia creates a stream that travels quite a distance, whereas with the mask on, that inertia is reduced and those larger particles are also captured. But what you also see is that it's not like it captures all of them, right? So these aerosols escape into the room, but they don't travel that distance. And that's why when combined with physical distancing, these masks are incredibly effective. Just to show you some data, so excuse me, our preliminary testing we did without masks. So we wanted to understand the impact of distance on travel. And so that's what you see here is we did it head to head and 45 degrees and 90 degrees just to see what the impact of different angles were. But the primary data here I want you to look at is the blue, because you can see just with distance, the number of particles decreases dramatically. Now you also see on here, we have some pretty large error bars, and that's because these particles are really fairly dramatically impacted by airflow in the room as well. And that's something that's kind of hard to control. So when we look at this, we do have big error bars on that data, but even with the big error bars, you can clearly see the decrease simply due to distance. And then when we throw in masking on the source, all right, we have just absolutely incredible results, even at 1 foot. the difference is dramatic. And again, this would be with those 2 micron particles. So if we, excuse me, zoom in on the data a little bit to see the difference between having the source only masked, having the target only masked, and having both masked. What we see is again, here's the reference level. So this is no mask, and this is the 1 foot comparison. So if we put a mask on, you can't even hardly see the counts that we're receiving with the mask on, the source mask. But this is the target mask. And so the target mask with the different types of masks at 1 foot, we're still getting a fair number of particles through those masks into the particle generator. And so, you know, that's pointing out very clearly to us, the source mask matters way more than the target mask. Now, it's still clear that the target mask does something. It looks to me like if I'm looking at this data, you get nominally a 50% improvement for the target mask. So that's the good, but this just reinforces the point that says my mask protects you, your mask protects me. And here we can zoom in on the data as we move out and incorporate distance into it. So this data is taken at 3 feet, and we've changed the scale because distance really reduces the amount of particles. So now the scale, instead of going up to 100%, which the previous one does, it's zoomed into the 20%. So you can start to see, but you can still see that the source masking is much more effective than the target masking. And of course, both masks work better, but the primary need is the source mask. And now this is the results at 6 feet. Since we had this setup in place, we also decided to run a 15-minute lunchroom test. So at Mayo, we're very careful around patients, and we've had almost no patient to staff transmission since we went to universal masking in the spring. However, our staff let down their guard in the break room, and that's been a real problem for us. So we wanted to set up this test system with no masks and count total particles, because our staff tend to feel safe, and they run into the break rooms together and they sit down and have lunch and they take their masks off and they pull the chairs over instead of staying far enough apart. We wanted to show them what was happening. So we took this and ran the test, normalized it to the no barrier state, which is our 100%. With a barrier in place, we were able to show dramatic decreases in the number of particles that the other person was exposed to. And if we added HEPA filtration to the room, it got even better. So it proves clearly, you know, some of the value of the barriers. And there is some question in some people's minds about how well the barriers work. And I think, like the masks, what they're working on is the larger respiratory droplets. They're really not going to do a whole lot for aerosol transmission. So conclusions that we came to: 1, masks on the source work significantly better than masks on the target. Masks on the target do minimally reduce particle count, so they do help. We found, this was one of the goals of the study, we found that the cloth and disposable masks we typically use here in our healthcare center are not statistically different, and that was really good to know. We also found greater distances in particular reduce the particle count, and it was nice to confirm that as well. There is one caveat to this, and it's something that as clinicians, you probably understand better than the engineers do, but these experiments do not reflect risk of infection, because we don't know how many coronavirus particles it takes to infect someone. So if I can reduce the number of particles you're exposed to by 1 or 2 orders of magnitude, okay, I know that's good. But I don't know if that reduces your infection risk, because if it only takes 1 particle, then it probably doesn't help a lot. And so that's something that is not well established. It's something our infectious disease physicians are talking about all of the time, the fact that we really don't know what the minimal infectious dose is. And so this is a study that shows that we can reduce the number of droplets you're exposed to. We are making no claims on whether that leads to, you know, less less infection. But I think anecdotally we have, and through other studies, we've seen that places that go to universal masking see the transmission of infection go down dramatically. That R0 number goes well below 1 when people are using masks 100% of the time. I'm going to close with just a quick video here of from within our engineering group, because this gives you a little bit better sense of some of what our group does and some of the tools and capabilities we have. Just a quick 2-minute video. It starts in our machine shop and then move into some of our lab space. We got a full machine shop, right? Your CNC mills and lathes. This is where we make a lot of the surgical tools that we develop. This is a femtosecond laser. So when I showed you the diamond electrodes earlier, the femtosecond laser was what we used to load the Girkin coating in the diamond electrodes. That was what you just saw was the reactor where we made those electrodes. Here you see a physical deposition system. This is what we use to create thin film electrodes. For example, the ones that we're using for our epilepsy studies, we need to create custom electrodes. Again, back to our machine shop. Create cooling area. We started working on some implantable technology. So this is some early testing we're working on that. And one of our favorite technologists is our glass blower. I don't know how common this is in Europe, but there are very few scientific glass blowers in the United States. The state of Minnesota, the Mayo Clinic, only has 2 of them in the entire state. So one of our founding fathers, Dr. Charlie Mayo, said, if we excel at anything, it's our capacity for translating idealism into action. In our group, our focus is applied engineering. It's creating these devices that can be used to help advance our practice, and it's an incredibly rewarding place to work. Let's see, I see that there's a question in the chat box. What's the ideal composition of a team trying to experiment with and implement 3D technology in a hospital setting in a low-income setting? That's an interesting question because 3D printers at this point are so easy to use that you don't necessarily have to have engineering staff. It depends on what you're trying to do. For example, our simulation center at Mayo does its own 3D printing for some of the devices they're trying to create to help train our physicians and our residents. Our group tends to focus on using 3D printing for engineering design. So in our case, you do have to have an engineering background to be able to do a lot of the design work that's needed. If we look at our anatomical modeling lab, which used to be part of our group and now is a separate engineering group that develops some of these custom models. Again, they've got biomedical engineers, but they also need the clinical side to help with the segmentation and to make some of the clinically related decisions. So from that standpoint, it really depends on what it is that you're trying to do. If you're trying to do some simple mechanical designs and develop some prototypes of new surgical instruments or something like that, often a relatively inexpensive 3D printer can do a lot for prototyping. But making finished devices, generally you start to need a little more expensive equipment. You need 3D printers that work with more engineering suitable material choices. But I do think there's huge advantages for 3D printing. You think back about the Hero arm that I showed you. That's made with inexpensive 3D printers. They're probably $3,000-$4,000 printers that they're using to make most of that device. A $700 device. So you don't need a high-end, $500,000 printer to do some of that kind of stuff.
That was brilliant. I mean, obviously, there's a reason why you are chairing the engineering side at the Mayo. We have lots of questions. I have lots, but I'm going to, for once, not ask a question.
I'm sorry, I've just lost my audio. I'll get it right back here.
Sure. Can you hear us now? Are you able to hear us, Mark? Or? Can you hear me okay, Mark?
There we go. Sorry about that.
No problem.
My audio cuts out every now and then.
Sure. Can you hear me now?
Yes, I'm back.
Okay, perfect. No, so I was just saying there's a reason why you are chairing the engineering developments at the Mayo Clinic, because you know exactly what you're doing, and it's incredible work. We have Maria and Chloe. They are our moderators for the conference. That's going to field more questions. So I'm going to hand over to them if you have time. Maria, Chloe?
Absolutely.
Thank you so much for talking us through all the recent advancements that happened in Mayo throughout the pandemic and all the recent advancements in regards to 3D printing. Definitely, we are living in a difficult era, but at the same time, exciting innovations are coming out. And my personal question is, when do you think 3D printing will be able to be implemented in every hospital across the world?
I'm sorry, I missed the last half of that question.
Sorry. When do you think that 3D printing will be finally implemented in all hospitals across the world? How far are we?
Yeah, so that's a good question. In the United States, and I can't say across the world because I'm not really sure about reimbursement in particular. One of the big challenges in the U.S. is until recently we really didn't have the ability to get reimbursement for the 3D printed anatomical models, for example. At Mayo, we've been doing those since 2006. And we've had no reimbursement codes for them. So we're doing them because we get better outcomes. You have to be willing to make that kind of an investment. And I would say that some of the hospitals in the US that are operating with very, very small margins simply can't afford to do that. And so what has happened recently is we finally got the reimbursement codes so that our insurers and our Medicare government systems will start to reimburse them. And that is what enables some of the small and medium-sized hospitals to start to implement some of this technology. So I really expect to see, most places that are doing complex surgeries to start printing 3D models over the next 3 or 4 years at this point, now that we can start to get reimbursement. I don't know where that is, if you look in Europe, for example, what reimbursement looks like. But again, in the US, outside of the major academic healthcare centers like Mayo Clinic, Johns Hopkins, Cleveland Clinic, some of those, many of them are just kind of operating with such low margins that they can't afford to do that at this point.
Sure. Chloe, any questions?
Thank you so much.
Yeah, so it's quite interesting to hear that obviously there was no statistical difference between cloth and disposable masks. But in the UK, we're currently only using disposable masks in the hospital. Do you think that potentially there could be a chance that we could then switch to using cloth masks? So then that would reduce any excess PPE that we're obviously using by constantly using disposable masks every single day.
Yeah, well, I'm pretty sure in our clinical care areas that we're continuing to use disposable masks. The reason we use the cloth masks is, we have 70,000 employees, and many of them are non-patient care staff, and we haven't wanted those staff to use disposable masks because there have been shortages in the supply chain. But the reason we're using disposable masks is because we want to throw them away between patients and between procedures, and I don't think that's going to change. And if you switch to cloth masks, you lose that ability. Now, if I'm working in engineering in the lab, if I'm working in finance or something like that, it's probably okay. I'm wearing the same mask all day, as long as it's not getting saturated. I think we will be sticking to disposable masks. I expect most places will. But it was very reassuring to know that our staff are protected, equally, nominally, no matter which type they're using. And there's also been a lot of talk about the bandanas that some folks are using. We tested those as well. They're very thin material typically. They don't work as well as a good cloth mask or disposable mask, but they're pretty effective also. Because again, what they're doing is they're reducing the inertia. So even though more of the particles are getting through, those that get through are moving slower. And so they don't go as far through the air.
Perfect.
Thank you for that. Yeah.
Mm-hmm. Sure.
Yeah, I did want to actually ask a question. I can't help myself. So I know that the Mayo Clinic has now moved over to London. Is there a plan for the developments there to have a combined clinical and engineering focus, or is it going to go on a different tangent? Do you know anything about the developments there?
Yeah, so it's a good question. So Mayo, first of all, does have an international strategy. We are, we live in a digital world now, and there's no reason why expertise has to be geographically constrained at this point. So we've got work going on in the Middle East. We've of course got the work going on in London, and are looking at expansion into other places through partnerships. Now, the partnership in the UK, I believe, is with Oxford, and I don't know that there's an engineering component to it right now. I think it's primarily clinically focused, but I think that's where our strength is as an organization, is in clinical practice. So it makes sense that that's where we start. But both in relationship to the UK partnership and with the work we're doing in Saudi Arabia, we're also looking at whether there's any value in creating engineering-based partnerships as well. The type of an engineering group we have and the capabilities we have is really, really unique. And it's a model that other folks could be interested in. If you think about it, most academic healthcare organizations do have biomedical engineering departments. They got people doing research. Some of them are making new devices, but their focus is typically grants and publications. We're not like that, right? We're applied engineering. You come to us and ask for something and we give you a device back, right? We're not doing research. We're not, focused on getting grants. We're simply here to provide new and novel devices. So it's interesting, and that means we have really, really strong expertise in quality systems, so that we know we're making safe devices, and our safety risk management processes are extremely safe. We understand biocompatibility and sterilization and things like that. And you don't find that kind of expertise typically when you go into a research space.
Absolutely. I think I need to speak to you more, because every time I have an idea for a device, it gets knocked back. So I think it should just come straight to you. We do have a few more questions if you have time. Chloe, Maria?
Mm-hmm. So one of the questions that we have is from Mahin, and she's asking whether recycled plastics are being utilized for 3D printing, and whether they can be implemented in low-income countries to minimize the cost further.
Yeah, so it's a great question, because Mayo and other healthcare organizations are part of within the United States, are part of a very large organization of green hospitals, essentially. And there was a large initiative started almost 20 years ago by President Bill Clinton, and many organizations signed up to it, and Mayo was one of those. So we have been making a very concerted effort of reducing our CO2 profile, our amount of energy used through more efficient HVAC systems, and trying to reduce our waste stream. So we're much more focused on recycling and things like that. But in terms of 3D printing, we're really limited based on material choices. So it's we're kind of stuck using the materials that the manufacturers provide, and so for the most part, for example, we make the 3,000 anatomical models that I mentioned a year to guide the surgeries. Rather than recycle those, what we typically do is we give those to the patients, so they can take it home and they can show their kids, show their grandma, So in a sense, it's not wasted. We're not throwing it into the waste stream. But I do think that there are opportunities for more recyclable materials for 3D printing. The challenge gets to be the material properties, because you have to have the right kind of tolerances, stiffness, all sorts of different constraints.
Absolutely. I'm probably going to have to be the bad guy and close the session at this point, Mark, because we're just going to bring in our next speaker for 4 o'clock.
Yeah, no problem.
Brilliant. And I would say to anyone in the audience, Mark, I'm sure you can be contacted directly. So if they have lots of questions or they want to collaborate, then I'm sure he will fulfill that. So thanks once again, Mark. It's been a pleasure.
Absolutely. Thank you for the opportunity. I really appreciate it. It's a lot of fun.
Incredible work. All the best.
All right.
Take care.
Yeah,
Thank you so
take
much.
care. You bet. Thank you. Bye-bye.
Okay, everyone. So we're just going to take a short break, and we'll be ready for our next speaker at 4 o'clock, so in a couple of minutes. So we'll be back shortly. Hi, John. Can you hear me? Are you able to unmute your microphone just as a quick pre-test? Wow, perfect.
I'm
Okay,
here.
fantastic. Yes, great.
Panic
Well, it's been insane so far in terms of there's been no technical issue, which I'm surprised about. So, and we're running to time.
I've been involved in running, was doing one with, actually the worst trouble was it was on Vancouver time. So I was chairing a session at midnight my time, which wasn't great, to be honest. But actually that ran, to be brutally honest, all right. One person it did fall over, but we've got video backups. But no, it seems to work actually.
Perfect. Okay, everyone. So we're back again for our speaker, Professor John Girkin. So we've gone to the United States in the morning, back to the UK, back to the US, and now back to the UK again. So anyone that knows anything about engineering, I'm sure knows about Professor Girkin. Massive experience in terms of optics and imaging, among other things, and also leads the way in terms of the EPSRC, heads up the organization around the technology side. And I think that's quite important. We have lots of ideas in terms of how we can advance clinical care, terms of technologies, but not all those ideas are gonna make it. So I think understanding that process and making those decisions are definitely tough. And I think this will be an awesome talk. So I'm going to pass over to you, Professor Girkin.
Right, thank you very much. If I choose that one, we should be in business.
Yeah.
Right. So yes, I'm John Girkin in the University of Durham, and because we've had Americans, Durham, UK, not Durham, North Carolina. And I'm going to talk about some personal experiences. As Neil said, I also chair the EPSRC Strategic Advisory Team, and I also, within the UK, sit on the MRC Translational Research Group, and I may make a few comments about that at the end. But as is the standard caveat now, any comments I give are entirely mine and don't represent any other organisation. So I thought I'd start by giving a little bit of a personal philosophical approach, and then I'm going to pick out two areas where I have done and we still are doing some work. And hopefully the philosophical approach will come out through that. And then there's a summary at the end. I should also say at the end, there's also a few questions that I've sort of posed that might go back in both directions to make people think. So I thought I ought to say, first of all, I'm a physicist. My PhD, as it says there, was in high resolution laser spectroscopy of atomic hydrogen. So very, very pure physics in that sense. And I then went and worked in industry for 4 years for a company that's photon controlled, and anyone that still uses some optics and optomechanical things. Some of the devices I made then are now sold by an optics company called Thor Labs. So the items down in the bottom right-hand corner, bottom left-hand corner, are micro positioners for fibers that were designed several years ago, and basically they are still in the Thor Labs catalog. And I sort of drifted into, if you call it biomedical engineering or biomedicine or biophysics, slightly by accident. And I went to work for an ophthalmic company in Windsor, UK, called Keeler. And I helped develop the puff tonometer. So if you've been to an optician and had a puff of air to measure the pressure in your eye as a marker or symptom of glaucoma, I have a level of guilt there. But also I fundamentally worked there and developed the diode laser systems for treating diabetic retinopathy, which is the instrument on the right-hand side, and also for use actually in surgery. So it's not a real premature baby, but one of the applications for that was using my device on premature babies. And I might come back to that in a little bit at the end of some of the advantages that you get working in this field. But I'll come back to that right at the end. So I then, if you like, drifted back into academia and became one of the team leaders at the Institute of Photonics at Strathclyde University, which has since morphed into the first Fraunhofer Institute in the UK. And again, the focus there was the application of optics to help advance life science and clinical activity. And then I moved down to become initially the director of a new multidisciplinary institute down in Durham, the Biophysical Science Institute. So you can see a little bit about my background. I also thought I'd put a few questions here about multidisciplinarity. I know it's a very broad audience, and so therefore, I thought, well, I'd just put a little bit in here. I think crucially here, when you're doing multidisciplinary, we're applying our physics, our engineering skills in a medical thing, and it comes back the other way as well. Make sure you get the original question right. Find out what the user really needs. In other words, it might be somebody comes to you and says, well, I need an imaging device to image this. And you actually, the question should be slightly different. I want to perhaps diagnose this disease using some imaging device. It's a subtly different question, because then that comes back of, well, okay, I can make an imaging device, and then I'll go and speak to colleagues in computer science who will then say, well, actually, we can then process the data. But what we really want to do is we want to make, if we're going to process it, could you just add this feature to your device? And you sort of go, well, we can do that. And the clinician then says, well, if you can do this, you could add in this. So you build the project up to make sure you're answering exactly what the clinician or the end user wants. And clearly in that, there is no such thing as the stupid question. You have to be open. People have to build confidence with each other. I'm also a great believer in transferring or stealing technology from one area to another, as you'll come. And you choose the right tool for the task in hand. So for example, and this is not a clinical instrument, there are caveats and warnings here, you could cut your toenails with a hedge trimmer, but clearly it is not the right instrument to use. So that's choosing, yeah, it will cut things, but it's really be careful. What is it you're actually trying to image, measure, work on, and then build the simplest, most reliable device to actually do that. The other thing, this is a slightly high risk slide, but I'm assuming I'm fairly safe where I am in Durham at the moment. And I apologize slightly to if there are any clinicians, but please take this in the best possible way. But I genuinely believe what I'm saying here. Again, it's this multidisciplinarity. When you start, if you're new working in a field, you've come from perhaps a much more solid engineering and physical science background, make sure you get to know and understand their language. And for example, also, they know a great deal, but it's about a different subject, and their driver, quite correctly, and this actually follows on slightly from the previous speaker, their driver is to make people better. Our driver, perhaps as an academic or an engineer, is, oh, I want to make one of them and publish a paper and do something interesting. Those two things are not mutually exclusive, but they're not the same thing. And the other thing that comes out is make sure that you understand that all your data will not be perfect. You know, if we do a physics or an engineering experiment and we plot a straight line, if we get some sort of chi-squared or correlation coefficient of 0.995, we go, oh yes, it's probably a straight line. If it's a clinical thing, if you get 0.9, it's brilliant. 0.8. It's acceptable, and 0.6 I've seen being viewed as being, it might be about right. So that we have to understand those things as well. The other part is, and again, please take this in the best possible way, you need to sum up the AK ratio. And occasionally arrogance is inversely proportional to knowledge, but be very careful. Yes, I've got a clinician leaning on my slide here. It applies both way round as well. So it comes back to talk, listen, and put it together. Just as because I deal with optics, I thought I'd make a comment. Clinicians have always used their eyes. So actually, particularly physicists have often dabbled or played with medicine. So you might be aware Archimedes, I'm not sure whether he's a physicist or a polymath. So he was interested in many things. And he was interested in the eye. And he thought stuff came out of the eye. hit the thing and went back in the other way. So clearly this was, we now accept, is not right. So be careful. Physicists haven't always got it right. The other thing, many people may be aware that during the Black Death, Newton left Cambridge and went back out to Norfolk, and he did some experiments. He did some work with prisms, which I've at one point tried to repeat and think's impossible. But he also did some work on the eye, and he took a leather sewing awl, so a big leather needle, put it round the back of his own eye and wobbled the back of his eye to see how it changed his vision, and then recorded them. I don't suggest you do that at home. And anyone that knows anything about Young's slits, so Young of interference patterns, was actually a clinician, and he built a thing to demonstrate that when you change the focus of your eye, it doesn't actually change its shape. So he put a pin on the back of the eye that was sort of clamped relative to the front and got people to look at long things and short things. And if the eye was changing its length, the eye ball would have pushed on the pin and it would have caused the retina to sort of flash slightly. Again, please do not do these at home. The other thing, if you're a physicist, everyone knows about Helmholtz and coils and magnetism, but he actually patented the first ophthalmoscope. So this was in about 1855, 1856, and that's his patent. But actually at the same time, this person, and in a normal, if it was easier for audience participation, I asked the question, who's that? And it's Charles Babbage. And Charles Babbage also built an ophthalmoscope, which he didn't patent. And interestingly, if you have an ophthalmoscope now, it's still built on one of these two designs. The Helmholtz one has a beam splitter, which splits the light in two directions. Cavendish's one is a mirror with a hole in, and you shine the light off it and look through the hole, and they both work. And the other comment I would say is that if you've got a life science challenge, or particularly a clinical challenge here, and we add some photonics, you end up with a solution or a product. There are two ways to doing this. You may create some new technology, and we'll see this shortly. And the other part is you may actually take some known technology and modify it. So both of those are entirely sensible ways forward, and they each have their advantages. And as I think I'm going to hopefully explain in a moment, there are some disadvantages as well. The other thing, when we develop something new for the clinic, it's got to do all of these things. It's got to be robust and reliable. It's got to be simple to use, because you can't spend a very long time training the clinician to use it. And very often anyway, you explain everything to the clinician, and then it's perhaps a scrub nurse or somebody else that actually does it. So you've perhaps trained the wrong person. It's clearly got to be safe. It's got to be designed for the purpose. And a bit here, it doesn't necessarily have to be low cost. It's got to be cost effective. So if you can save overall, that's clearly a benefit. And that cost effectiveness may take place over a very long time period. And the other one is underlying it, it's got to improve the quality of care. That's why we're doing some of these things. So let's take an example. And a sort of novel approach to caries detection. So briefly, I explain what I mean by dental caries. So this is dental disease, and what we're probably aware of, we get a bacterial film, in this case green, and they build up on the surface, and they're eating sugars in particular, and they excrete an acid, and that slowly starts to dissolve the top surface of the tooth, the outer surface of the enamel. And initially what happens is it makes it porous. So it doesn't form a hole straight away. It's just affected the top few microns, and it's made it slightly porous. And at that point, the bacteria may stay there. And now what starts to happen is that top surface still stays there, but we now start to lose mineral from underneath the surface. And that's what's happening here. And at that point, in essence, two things can happen. One is, the bad route is that eventually undermined the surface of the tooth to such an extent the top collapses, and we then end up with a cavity, which is almost certainly going to need restoration, as a dentist would say it, drilling and filling, as a victim might say it. But there is an important point that at this stage, at this intermediate stage, if you keep the tooth really clean and you dose it with high doses of fluoride and some other materials as well, the tooth will actually remineralize, including the top surface. If it's cavitated, it's gone. But at this early stage, there's still a protein matrix inside the tooth, and that can give you protein-guided crystal growth, which in essence means that the tooth will remineralize. The trouble is, at this very early stage, the dentist is basically looking for a white spot on a white background, which is very difficult to see. And it's also at such an early stage that it won't appear on an X-ray. If it appears on an X-ray, it's probably gone beyond the point of remineralization. So important bit, it's reversible in the early stages. So you go, okay, this is what's happening clinically and biologically. How does that affect me as a physicist or a bioengineer? So I then look at the tooth, and what do I see in the tooth? It's made of an outer surface of enamel. And the dentist will call these prisms. They're actually rods. And they have a they're made of calcium hydroxyapatite, and the center has a very high refractive index, much higher than water, much higher than most glasses. And round the outside of that is a lower refractive index material round these rods or prisms, and they are about 4 or 5 microns in diameter. And to me, as an optical physicist, that says, well, that's not a bit of enamel, that's an optical fiber. And effectively, what happens then is any light that goes onto the enamel gets bounced down, as it shows on the right-hand side, and is guided through the tooth. And eventually, yes, it will hit the dentine and eventually get scattered back up, which is why your teeth are this sort of whitish, yellowish color. And clearly longer wavelengths are scattered less. The thing on the left is something I use as a demonstration. So it's a laser going down a water fountain, and you can guide it down, and it looks really pretty. So to me, the important part is what happens inside the tooth. When we get some damage, these early caries, it's going to disrupt the structure of my optical fiber. So the light is no longer going to be carried through it in the same way. It's going to be scattered back towards the user much earlier than if it had come from the dentine. And this is why the dentist then gets a white spot. It's light that's been back scattered towards them on this white background. So how can I help this? So initially what we built in the first version of this literally was a CD player with an 810 near infrared laser diode in it, and we shone it through a tooth, and we put a webcam the other side, and in essence you end up with an image that looks like this. So if you look on the right hand side, you can clearly see that the enamel is transparent at this wavelength. This is the enamel part, this is just dentine underneath. But here we have this gray spot because we're looking in transmission and the light's going basically through. And this is an area which is where the enamel has been damaged at an early stage. So this would look, and it's damaged. So the light in this case is not being transmitted through in quite the same way. You can also begin to see down here that you have a pulp chamber because the dentine is thinner. So we thought, well, this was quite a good idea. And it's really simple because all we've done is taken a laser diode. In the end, we used an infrared LED and a camera, and we built a clinical instrument. So this is the handpiece, and inside it there's some optics. The LED is mounted here. There's a mirror here which sends the image down here, and we had a reasonable quality camera stuck at the end. and you can take images through the teeth. And we did actually use this on some willing volunteers. We actually had a real dentist used it in my office, and we imaged a number of people's teeth. This again is an extracted one. This is where it was extracted, so ignore that. But in this area here, you can see a dark area. That was a very obvious lesion. The dentist, you can see that very clearly, even under normal light. What's interesting here is this little lesion on the left-hand side. Nobody had seen that until we looked in the infrared. And then when you looked afterwards on the tooth, you sort of thought, yes, okay, I can see it's a slightly different white color from the rest of the tooth. The same is also true here. This is actually quite, you can see this is an incisor, and clearly there's micro cracks, which would be relatively normal in this case. There was a proximal lesion where it was touching down the side here. So this was showing us and giving us some information, and we thought this was quite useful. But what people really wanted is, can we quantify this? Can we actually almost produce three-dimensional images? And what we did then is we looked at a technique called spatial frequency domain imaging, which has been used in tissue initially by a group. It was started in about the mid-1990s in this format in UC Irvine by Bruce Tromberg and his group. And we said, well, nobody had applied this to teeth. And the optics of a tooth, as you can see, are quite different from normal tissue. And in simplistic terms, what we do on the left-hand side of the slide, you can see we project a pattern of black or dark and light stripes. And as these stripes propagate through the tissue, they go out purely due to scattering, or there may be some absorption which removes photons. But as you go through the sample, the pattern is being blurred out by the scattering. And clearly, if you have a widely spaced pattern, that blurring will occur at a deeper depth than on the left here. We have a more tightly spaced, a lower spatial frequency, and this pattern clearly gets blurred out more quickly. So if we put different patterns of light on, we can now begin to get determine when the pattern has disappeared. We can get some information on depth. So in essence, this is what we have. We have our tooth, and we project a grid on it, normally a sine wave, but you can actually do it with a black and white grid, and we look at the pattern of light that's coming back. I should add, we deliberately put a polarizing element in, so it's backscattered light, not light reflected off the surface. And we take the pattern, we take an image, and we move the pattern 3 times. So there's once, and then we move it, and again, and then we move it again. So that if we just added those images, we would have completely even illumination. But if we add them in a different way, if we do the difference, the sum of the difference of the squares, what we then see is not just the even illumination, we see where the pattern of the illumination is. And we can then do this at different spatial frequencies, so a high frequency one or a much broader one, and we can do this at multiple different frequencies. And you'll see we can then begin to reconstruct. So we're putting on a modulated light pattern that we know is going to disappear at different depths. And I will accept that this is not a system that was used in the mouth. I'll come back to that in a moment, but this is the way we tested it in the lab. We have an LED collimated. In here, to produce the pattern, we steal what's inside a digital light projector, which is an array of micro mirrors, and the sample, the tooth, sits here, the light comes back, and one polarization is selected to then go onto the sample here. I should add that the clinical version of this actually has basically the same optics, but we go into the mouth here using a coherent fiber bundle, a multiple array of microfibers, which I'll talk about in a moment. But the microfibers take the image into the mouth, the image comes back and then goes onto a camera which is mounted in a base unit. So it is possible to do this. And on the left-hand side, there's the light projector, goes down into this polarizing beam splitter. The other polarization, in other words, the scattered light, comes back this way because the scattered light has its polarization altered, and it goes off into the camera. And we need to do a little bit of image processing. So if we think of it this way, I know what the light looks like that goes in. I know what the detected looks like, and I know that my pattern is going to degrade with depth. I can calibrate that either mechanically or you can actually do it mathematically. And we also know, as we saw earlier, that lower frequency, wider space patterns are going to come from deeper within the tooth. We remove the DC image, that's just the direct sum, and we process the others. And we can then look, if we've done this at multiple frequencies, at what the intensity is through every picture, every image. So we've taken the images, we've just got the alternating signal, we put those into an image stack and select one pixel that goes through that. The different images in our stack are the different spatial frequencies, and we can calibrate that, and we can then begin to create a 3D image. So if you have strong scattering, the photon goes in and comes back quite quickly, and the pattern's going to be blurred. Low scattering, it's going to take a longer path. And if we then look at our signal here, where we have high scattering, we're going to end up with something that looks like this. Low scattering, it looks like this. And the other part that we need to put in here is if we have something which suddenly has much higher scattering, it's going to reach a higher curve, and then it'll eventually return to this lower value at a certain depth. So if we've got something that's doing this, this is indicating that there's some high scattering that's taking place near in this case, near the surface of the tooth. And we can then plot these. This is a surface defect. This is different spatial frequencies. I should add this bit here is damage that was caused on the tooth, but there are some marks that are present here that are due to changes in the enamel, and we can then begin to see these. For those of you that have a dentally nervous disposition, there's no nasty mouth, but some pretty horrible looking teeth here. So let's use the one on the right-hand side first of all. So not joking about dentists, any dentist would spot that one. It's a huge, great big cavity that's formed in there. You know, even I can spot that probably needs a bit of treatment. The bit that's quite interesting is this one here is actually not a cavity, it's staining within the tooth. Because if we look here, there's a little bit of damage, but most of that is surface. So there's no lesion in there. That's actually relatively healthy bits of tooth. The bit that's quite interesting is this arrow in here. Actually, in this normal image, it looks not too bad. Actually, we can see here that there's a problem beneath the surface. It's almost certainly connected to this. If we look at this one, again, quite a big dark problem. This one is another lesion. The other bit that's interesting here, up here, there's obviously some mineral lost beneath the surface. It's a bit unusual actually in the top of the crown of the tooth, but we can see here that the mineral structure is not the same. The one that again here is okay, so dentist probably spots that one, but here that looks like healthy tooth. But when we look, and these are 3D images I've just collected together, you can see that there's a large loss of mineral just beneath the surface here. It's not really affecting our normal image, but it's affecting this one. And this is an area, if it appears like this, so it's hard to see visually, but it's underneath, that's exactly the sort of thing where remineralization would take place. I would accept that in this case, that one may very well need drilling and filling. And if we take this one, again, looks perfectly healthy. Actually, there's some early loss of mineral down here. This would have been where the two teeth are in contact. I should add as well that this image was actually taken this way. was actually taken this way through the tooth. We've just reconstructed it to look at it sideways on so it matches this image. But we can see here that where this tooth was touching the one next door to it, there is some caries, there's some early caries forming. So you tell the patient, keep it clean. You might give it a wipe out and you'd coat it with varnish. So this is an instrument that we're now looking to take forward and commercialize. It's very interesting. The only way to do this, to take it to the next stage, it's got to go beyond the lab. We need to build multiple instruments to do the relevant trials and to begin to get people interested. So we are looking now to raise funding to do this at a company scale, not at not carrying, so building from the lab, because that's the only way we can really get it into the clinic. So the second project that I'm going to talk about in the remaining time is an EPSRC project called Proteus that's run out of Edinburgh and is run, the clinician is someone called Kev Diwali, and the coordinator is a chemist called Mark Bradley. I am part of this project. There are also partners from Heriot-Watt, Bath and Dundee. So this was a project that was originally funded 2014 and is now building on. And overall, there are probably about 50 to 70 people that are involved in this project with PhD students and covering a whole variety of different topics, as well as postdocs and full academics. And what are we trying to do? So the idea is, yes, one can put a bronchoscope inside a patient's lung, and these are patients who are in intensive care. I should say this whole project was started well before COVID, but we actually think that our instrument, the first time it was used on a COVID patient, was the first time anyone had actually seen at a microscopic level what was going on in a living COVID lung. But so this was basically looked at not for cancer diagnosis necessarily, but very often patients can get a fungal infection in the lung or a bacterial infection in the lung. And on an X-ray and for multiple other diagnostic techniques, they look very similar. The crucial bit is, well, two things. One is the patient can get sick very, very quickly. Kev has a story about a very fit man in his 40s with a family that came in. He was a regular fell runner. And so he was a very fit person, came in and had a chest problem, which got worse. They sort of kept him in to diagnose him, and he kept him in overnight, and he was transferred to intensive care, and they could see the problem in here. And unfortunately, in this case, he died 3 days later. One of the challenges is, if the patient has a fungal infection, you would give them one type of drug. If it's a bacterial infection, you're likely to give them another type, but very often the antibacterial compounds are actually growth promoters for fungal infection. So you have to be careful. So the idea is, down a normal bronchoscope, can we put a very small imaging fiber, which is made up of multiple individual single mode fibers. We scan across that and image what's going on inside the lung. And this is a whole thing across here is about 1.3 millimeters. We may also put some fluorescent beads or detectors to look for what the chemical aspects are in the lung, and a hollow thing here so we can either squirt some fluid in here, or in the case when, as I said, this was used on some COVID patients, we actually put down compounds that they thought might help, and we could put a very small quantity onto the viral infection in the lung and actually see if it made any difference. And the dose was so small it wasn't going to cause any problems elsewhere. So what are the fibers? And these are the same sort of thing that we're going to use in the dental imaging thing. These are made by Bath University, though you can buy a commercial one. So on the right-hand side, they pull an array of optical fibers. So those of you who don't know, to make an optical fiber, you take a large chunk, round block of glass, and you pull it. So what they do here is they stack multiple blocks of glass with slightly different diameters. They pull it, and then you cut the pulled bit and you stack that bit on top of the other one, and then you pull it again and then you stack it. And then in the end, you end up with between 8,000 to 30,000 cores that are all between about 3 and 5 microns. They're quite deliberately made different sizes, so you minimize crosstalk between the fibers. The outside diameter is about 1.3 millimeters, and there's a 250 micron catheter. And then sensing fibers, these are individual fibers where the end has been etched, so we can put sensors actually on the end, which it all goes down into the same thing. Mark Bradley and the team of chemists, their main expertise is to develop novel chemical markers. So these are fluorescent sensors or Raman enhanced sensors for detecting different things. So these may be sprayed into the lung in a local area to label specific types of bacteria, or they may be to sense the local environment. Crucially here, as you'll see in a moment, these sensors both have different wavelengths, but they also have different fluorescent lifetimes, and that's an important feature. And the first version of this was used one of these fibers. This is a USAF test target. And we just used 2 LEDs. They went down through a microscope objective into this small fiber bundle, which was inserted down the bronchoscope tube, comes back. And we then, by turning on the red LED or the blue LED, we can then look at different wavelengths and detect this on a color CMOS camera. And effectively, this is the use just before it went into an intensive care unit. This is Nick, who's now at Dundee. And this is Kev, who is the clinician, Kev Diwali, who is the clinician involved. And we can see inside the lung of this is from a live patient where we have some bacteria which have been labeled fluorescently red, but we also have this green material, which is fibrotic tissue in the lung, which is causing a problem. You can also then go down to these individual neutrophils, very relevant for COVID, and there are monocytes, and we can now fluorescently label them in different way. And this one here, item C, actually has fluorescently labeled to fluorescently label a fungus that's present in here. So this was working, and there are quite a few of these units now around the world, in Europe. And we then went and actually we also wanted to know, we'd like to measure the fluorescence lifetime. So you put a short pulse of light in, how long does it take to go down? And a new level in the last 10, 15 years of detectors called SPADs, single photon avalanche photodiodes, which are based on CMOS technology. So they were originally designed for going into mobile phones. And can we use this CMOS technology to build the devices that we want? The other bit that I think is important to know here, that you can actually, through a thing called Europractice, buy space on a semiconductor wafer, and it's not very expensive. It's about £3,000 per square centimeter. So and for that, you would get typically 30 to 50 areas that size. So they would do 30 to 50 wafers, and so you've got perhaps 30 to 50 of your little chip on it. So there is a way that you can do this and put it on. It's part of silicon photonics. And the crucial bit here with these detectors, they're very small. And what happens, if I look on the right-hand side, a photon comes in, it creates an electron, but the actual thickness of the device is very short. It's 2 or 3 microns. So if we put about 10 volts across it, that electron really gets accelerated very rapidly. So one electron hits another electron hole pair, which creates 2 more, which creates 2 more. So you get this avalanche, this breakdown. So a single photon can give quite a large signal. And because this size and everything is so small, the time it takes for the photon and electron to go through the system is very, very low. So we've got a very fast detector. So we can then use this technique called time correlated single photon counting. So we have a fast detector, we put a short pulse of light in, which starts a clock. And then when a photon is detected, we stop the clock. So on the right-hand side, the first photon arrives here, the next pulse, it arrives here, the next one at various different points, and we can then sum all these pulses up, and we build up, in essence, an exponential decay curve, which is the fluorescence decaying, and we can do all this within our chip. You need high-speed electronics, but we can actually put, because this is CMOS, we can actually put all the electronics on the same chip. So this is the detector. In the end, it's 512 pixels across here. A pixel is a pair, and they're actually 8 pixels high, but so this thing here is a single detector, and we can either take the signal from any one of these from the top or the bottom, and the red ones are slightly more red sensitive. And we can do normal counting with time correlated single photon counting, all integrated on the chip, or we can put all the detected photons in individual buckets or bins, and then do some very simple things. So you could literally have 2 or 3 time bins. These are the early photons, these are the late photons. And as you'll see, this has now enabled us to do real lifetime imaging inside the human lung at about 15 frames a second. These are actually the detectors. This is what they look like in the end. And this is the way the system is built. So here is a short pulse laser source. A continuum laser goes into the optics or into some dichroic optics. There's some scanning optics in here, which is what we designed in Durham, which is all reflective, because reflection, nice thing about reflection is it's achromatic, so it doesn't affect the wavelength. And the light goes down here, and you can either put a sample, or this is where we now put our coherent fiber bundle to image across the fiber bundle. So we can image each fiber individually. The light comes back, goes through here, and we then have a spectrometer here, so that for each pixel, each point we've scanned, the return light gets split, goes onto our detector. So the array of pixels now have blue detection at one end, red detection at the other. So we've taken our spectrum and divided it into 512 wavelength bins. And for each one of those bins, we can measure the fluorescence lifetime at that wavelength. And we can do this very, very rapidly and pull the data off. The biggest problem here actually is if you think that you've got an image that might be 256 pixels by 256 pixels, and for each one of those pixels, we have up to 512 wavelength bands. And for each one of those 512 wavelength bands, we can have up to 32 temporal bins. The data gets quite big, but we can pull this off very rapidly through a USB 3 interface. This is the way the optics were done. And this again is an engineering challenge. So this is going to have to go in a clinic. It's going to get bumped around. So this was made, the optics are in there. You can see the light comes in. Here are the mirrors that were shown previously. The scanning galvanometers are there. This is the output which goes down to into the patient. And this is all made from a solid block of aluminium that's machined out, and then the optics attached to it. It makes it a very rigid structure and also helps with thermal stability as well. And then what do we get in terms of the images? So this is this is not medical. This is a plant, convallaris, but it works quite nicely. So this is now binning some of the colors. So on the left-hand side, this is all the colors that were red. This is all the colors that were blue from the fluorescence. But this one here, so we can add those together to get our color image. But then for each pixel, we can then look and plot what the fluorescence lifetime was, as well as the wavelength. So we can actually look at the color. So the image here on the right-hand side now, this is not an intensity color coding. This is a lifetime coding. So blue is very short, red is longer. So this is for this image here, or this is now looking at it in terms of lifetime. So you can see this stands out very well, and it's at this wavelength. This bit here doesn't really appear very much in the image, but clearly has a different fluorescence lifetime, which can tell you a great deal about what's going on. We can then look at them in a different way. So this is actually from a piece of a biological tissue, and we can then look. So this is the intensity profile for a particular pixel. And then we can also take a slice here. So we could then take this wavelength for the whole pixel the whole image that's been scanned across, and plot the lifetime. So this is the lifetime at that point. This is the lifetime at this point, and that's what the image looks like. So we can then begin to break up these spectra to try and extract what's important. A bit that's important here in the human lung, that fibrotic tissue that I mentioned has a very different fluorescence lifetime from more normal natural tissue, which is also very different from any chemicals we may put in. So we now have a simple way to remove the fibrotic tissue, or image it, but store it in one place, whereas the other bits that are of interest we can get elsewhere. This is some ex vivo cancerous lung tissue. Not going to go into the detail. This was actually H&E stained, but we can actually do it without the staining as well. And there is a clear marker as you go across that the fluorescence lifetime has a slight, the fluorescence is a slightly different wavelength, but an even closer marker for malignant, or sorry, in this case, I should say cancerous tissue, was that it has a very different fluorescence lifetime. And we can take these images, as I said, we can take a 256 by 256 image at around about 15 frames a second. This again is an image taken down an optical fiber. This is only fluorescence. This is a raw image. You can just about see some of these little dots that you can see in different places. They're the individual fibers. We can smooth that out. The red here are bacteria that have been labeled in one color. The green again is the fibrotic tissue. So I've given you a really quick idea. This is a system that, as I said, version of this was the first use of the beam scan system in real humans was on COVID patients in April. It manages to speed it through the process. I was going to end by just making a couple of comments.
Sure.
When we develop new instruments, these are some of the challenges we've come across. When I developed the pulsar, the puff tonometer within Keeler, basically one of the things that came out was the accepted clinical gold standard was faulty. So this was a standard that had been around since about, I think it was 1930. And basically our instrument was trying to calibrate it with a standard that it became clear was faulty. That causes a few problems. Actually, clinical practice has now changed, and they now measure the thickness of the cornea as well as measuring the direct pressure. So that's an important point. The diode laser photocoagulator was much cheaper, much more convenient. You could carry it around in a suitcase. And we targeted a certain price. Actually, the price was too low when it first came out. Sounds really odd, because people thought, oh, it can't do as good a job as this expensive one and be that price. So we actually sold more when we put the price up, which is somewhat counterintuitive. The dental imaging, very difficult. We found it, as I said, very difficult to fund it. And the last part is taking the thing into the clinic, particularly with the chemical compounds. And the previous speaker from the Mayo Clinic touched on this. It's very difficult to take an instrument I may develop in a physics lab and get permission now to get it through and used on patients. This is a real challenging place, and it's not being well addressed. Hopefully, I've given you a bit of an idea of my philosophical background. Some novelty in dental detection, where we clearly used things that were available off the shelf. Proteus, we've really pushed things further forward. We've developed really quite novel things as we've gone through. The SPAD detector was inventing new technology specifically for this. And I've given you a little bit of a hint on the challenges. The thing I must quickly on the dental things, the last two people I've been working with here, there have been people in the past, but it's Nigel Pitts and Alistair Bound. So Nigel is the dentist. And then in the Proteus project, crucially here, I must mention Mark, who runs the project, and Kev, the clinician, Robert Henderson, who's the man who invented the SPAD detectors, again in Edinburgh, Gareth Williams, who actually built the system, then down in Bath, it's Tim Burks and Jim Stone with making the fibre, and Nick and others in Dundee. I cannot cover everyone, but I have to thank those people. Right, there you go.
Fantastic. It was brilliant. Absolutely brilliant. We do have questions, so we do have question moderators. So I'm going to introduce you to Luxie and Laura, who are based at Imperial College London, and also Faisal, who's coming in from Dubai. So if they have any questions, that would be great.
Right.
Laura, can you hear us?
Anyone want to open up?
Yeah. Hi, Professor Girkin. Hi, my name is Lucy. I'm a PhD student at Imperial College, working on optical sensor. So I'm I think your presentation is very interesting and very relevant to my background. So from your perspective, what would be the potential applications for optical fibers in areas other than what you have introduced, such as dentistry and lung and cancer?
So I think there's, I'll preface my answer by saying the comment, again, it comes back to my choosing the right tool for the right job. And therefore, so I think the advantage of the multi-chord fibers that we can now make, or they now make very well in Bath, and Fujikura can make them, is that it does mean you can do minimally invasive imaging at very high resolution. So the one comment I didn't make, the images that were there all have a spatial resolution of around about 2 or 3 microns, which is good for some things. So if you're trying to actually begin to look at cellular level structure, which may be important, it's very definitely important in cancer diagnosis, for example. That's what these give you the advantages. We've also got ones where we've put miniature graded index optics on the end of it, which gives you a slightly wider field of view. So I think in terms of imaging, it's probably looking for high resolution. I think the other part that it's worthwhile remembering, though, whether this is the best thing for them, you can also embed sensors either on the end, or you can use some of the properties of the fiber to measure temperature and other things like that. So I think the other side of it, which we're beginning to use here, is again for treatment, for light activated treatment. So the idea that we could use a photodynamic therapy, which may be cancer related, and I'm working with some chemists in Durham who've made small molecules suitable for this. And the crucial bit is we're not just putting them in and then illuminating them. We're looking to see how their spectrum changes and how their lifetime changes, because that then tells us a great deal about, is it killing the cells? What routes it's killing them? So I think at the moment, some of them are clearly for diagnostic. I think a lot of it is going to be to actually understand what's happening in vivo. Because what happens when you look on an in vivo sample, it's totally different from if you've got a selection of cells on a plate. And I think so, therefore, there's 2 sides to that. One is thinking of it for in vivo clinical diagnosis. That would be useful. But I think the other one is, can this actually give us a better understanding of what happens in a real patient, rather than a cell line that might be sat on a cell bench, on a laboratory bench.
Absolutely.
Yeah. Thank you. Thank you, Professor Girkin. So I have another question, is that as we know, we have so patients with these kind of disease are kind of like people with disease, the disease are long term. So in terms of managing the disease for a period of time, rather than just take the optical fiber, do the imaging or sensing at the hospital settings, how do you think it's a promising area that we could potentially put the fiber inside inside human body for a long period of time?
So technically, there's no well, if it's a glass and you coat it with the right polymer, there's no reason why the body would reject it. So glass, and you could even, and I mean, intraocular lenses for cataract surgery, they're PMMA, they're plastic materials, and they survive perfectly well in the body. The body doesn't actually react to them. So you can minimize any response that the body might have. The slightly bigger issue sometimes then is, well, if I need to get light into my fiber and light out of it, you may do that, or you may say, well, actually, we can encapsulate at one end of a fiber that we've embedded in a patient. We can put in a very low power LED. We can put in a very low power detector and the electronics. That can all be then sealed in the patient to come out. So there are people who are using heart pacemakers now, where they're actually stimulating the heart with a light pulse that goes in and stimulate. It produces electricity, which stimulates the heart. And that has found to be better and longer lasting sometimes than putting electrodes onto the heart in a conventional way. So there are ways that you could do it. The problem is that you do really need to put the light source and the other thing inside. If not, you're going to end up with a bit of glass sticking out of the side of your body and some LED, and that's not going to be a good idea. But the point is, you can use low energy electronics and things that you can actually then embed them, and they will last a long time inside the body. So that's probably the route to go down.
Okay, thank you very much.
Laura,
Brilliant. Sorry. Yes, hi. My name is Laura, also from Imperial College. Thank you so much for your talk, Professor Girkin, and also for your answer to the question so far. It's been really, really interesting. We have one more question that's come in from the audience today, if that's all right.
Yep.
Yeah, so someone's asking, how challenging is it to communicate discipline-specific concepts to other members of the team and work cohesively?
Great question.
That is an outstanding question. I think, well, okay, I'll put it two ways. I think there are some people that can do it well and some people that don't. So I would put that as a caveat at the beginning. I think it does come back to using the correct sort of language. So my joke slightly that clinicians know a great deal, but it's about a different subject or a different language. So when you explain something, and this works both ways, you don't want to dumb it down, but you need to explain it in concepts that would be that people are able to understand. And crucially, it's listen to the response, and you can normally get, did they understand that? No, they didn't quite understand. Right, I'll explain it in a different way. So I think it's asking the question, and very often the way that I will do it, you need to build up confidence when you first meet people. And I'm more than happy to ask, inverted commas, the silly question, because generally, and the other thing is you need to get people, if I don't know the answer, the trouble is there's lots of other people that don't know the answer that I got, and the same works back the other way. So once somebody breaks the ice, you can then begin to talk it through. And it's to use, yes, don't make the language really very, very simple, but use terms. As you may have gathered from the talk that I've given here, that I do some of this with a certain sense of humor as well. I think that helps. But it's you don't dumb it down, you use things. The other comment I would actually make specifically, and this happens a lot with clinicians and life scientists, and this was told to me by the senior cardiologist at Harvard Medical School about 3 years ago, who I was working with. And if you're worried about how much funding you get, was it Harvard Medical gets $7 billion a year for funding. $1.5 billion goes to the cardiac unit. There you go. That's the level they're playing at. He made a very interesting question, which is very often, and this was his case as well, people become clinicians or they may become biologists because they really love science, but perhaps their maths is not as good as it might be to be a physicist or an engineer. And if you think about it, in the UK, many biologists or clinicians will have done very little maths beyond, sorry, I'm GCSE. And therefore, there are certain concepts that you have, it's things like that that you need to remember. Yeah, they'll know an awful lot more. And it comes back the other way. So be careful how you explain it. Think what level of training somebody's got up to, and then build on it from there. So, you know, if you instantly went out and said, oh, well, I did a linear regression on this and it has this and this sort of thing, you're probably going to lose the person. It's putting it in the right context. I should add one other comment. When I started working with clinicians in ophthalmology, I used to have a real problem because they talked about anterior and posterior. Anterior pole of the eye, it's the front and the back, but they use, and it's the same with left and right. So be careful, don't use acronyms, use everyday words.
Absolutely, I fully agree with you there. Faisal, anything from your side to close?
Right. Yes. I mean, Prof. Girkin, it's been amazing listening to your entire talk. I think it was very comprehensive, yet to the point for something that covered each area of impact that you've made, which I think is impressive on its own. I'm interested to really understand, and you mentioned, and you covered this earlier in your presentation about how much history has played a role in bringing us to the innovation that we have today. And I'm curious to know how much has it influenced you and your path in choosing to impact these areas of science?
That's an interesting question. Again, I'm going to answer it by caveating. I have never really planned my career. I did physics because, well, I'm quite good at physics and I'm not that good at, I'm competent at maths. I'm not a mathematician. So I did physics and, oh, actually, I quite like lasers and light. So I quite like to do a PhD and, oh, okay, I'll do a PhD. And I drifted in. Having said that, it wasn't so much the laser or the optics or the physics of it. It was actually being able to apply it in something useful. And so when I went up to Strathclyde, I knew I wanted to do some applications of photonics in the life sciences. And the other thing at the time, I didn't want to do cancer because everyone was doing multiple different ways of imaging. So that's how I got involved in the dentistry side of things because, oh, well, actually, there's a lot we could do here. So it's finding something. Yes, I wanted to be useful. And if you like, what I like making, I say this, you know, what do you do? Oh, I make optical toys. And it is almost that's what I do, but I make them for a practical purpose. So it's looking at it. And yes, I'll end with this one comment. The greatest, I have to be careful because I'm in the house here, but one of my children's still around, but the other one isn't, and my wife is. Anyway, hopefully they won't hear me. Probably one of the greatest thrills in my life was one wet November Friday afternoon. And I took the laser that you saw earlier. I had a phone call in the morning from the London hospital saying, we have a premature baby that needs some treatment. So I got in the car, drove it down. It was a grey, wet, horrible day and I explained to the clinician how they were going to treat this premature baby. And the baby came in. And he explained to the mother how it was going to be treated. And she said, I'll have to leave now. And he said, no, with this laser, we can do it here. We don't need to anesthetize the baby. You can hold your baby's hand. And while the treatment's going on, I'm just standing in the corner of the room. It's got nothing to do with me. I should say I'm having to be careful now because I could start crying in a moment. And I'm not going to, but you'll see why it's so emotional within me. And they treated this baby. And the previous treatment had failed and with a different method. And after it had worked, the clinician came over and said, did I want to have a look? Because I knew what I was looking for. So I then had a look at the back of this baby's eye and went, I think that's worked. And he did as well. And so I then disappeared into the corner. And then the clinician said to the mother, you might like to know the person that invented that instrument standing in the corner of the room. And this mother just ran over and gave me a hug. All I can say is, as I said, I can now feel myself almost welling up with it. I didn't walk out of that hospital. I just floated all the way back down the London Road. It was incredible. That's what gives you the biggest kick. You know, that was quite a few years ago now. I don't like to think how long it is. It is still vivid enough in my mind that I can give myself an emotional experience. If you do that once or twice, that's it. That really is a really good reason why you want to do something like this and apply my optics skills. I couldn't rate the baby better, but I could help somebody else make the baby better.
That makes perfect sense. Incredible story. Thanks for sharing, Professor Girkin. So we're going to close the session now as we just prepare for our final speaker. So thank you once again, and we'll just get ready for our next presentation.
Thank you. Goodbye.
Thank you.
Thank you.
Thank you.
Laura, can you hear me okay?
Yes.
Perfect. I'll just let me just see.
Should I think our next speaker is in the attendees.
I will just okay. You are ready to go. Let's see.
Hello.
Oh, perfect. Okay.
Brilliant. Oh, great.
Hello. Okay, brilliant.
Hi. Well, you've promoted me. Thank you.
Okay.
Oh gosh, sorry. John's made me cry now.
Okay. So thank you for joining us, Professor Stride. So I'd just like to introduce you first. So our next speaker is Professor Stride from the University of Oxford at the Institute of Biomedical Engineering. Her academic studies in the past have included a Bachelor in Engineering and a PhD from University College London, and she is recipient of numerous awards and has published significant research studies on drug delivery systems and biomedical ultrasonics. So without further ado, let me introduce you to our next speaker, Professor Stride.
Hello, thank you very, very much. I'm sincerely hoping that's displaying. Can you see my slides?
Not quite.
No.
Are you able to maybe share your screen?
That should have been doing that. Hang on, let's try again. Oh, I've got
brilliant.
Oh, great. Can you see the screen? Right, let me go back to presentation.
Yes, that's working.
Sorry, it was playing up earlier. And oh, if I do that. Hopefully that's full screened.
Yes.
Brilliant, fantastic. Thank you very, very much. I was so enjoying John's presentation. And that there were so many overlaps. I'd also like to echo everything he was saying about the importance of collaborating, particularly between sort of the so-called hard sciences and medicine, and just doing our very best to make that happen. What I'm going to talk about today, I hope, is an example of how we've tried to do that. So what I want to focus on is a project that is collaborative between engineering science in Oxford, 2 hospitals, one in Oxford, one based in Belfast, and particularly a group in Northern Ireland at Ulster University. And we're all working together to try and develop a new therapy for pancreatic cancer. So it's a very large team. This is a selection of the people involved. So a lot of the data I'm going to show today were acquired by Josh Owen, Estelle Begin, and Heather Nesbitt. But all of the other people on these slides are absolutely crucial. John and Tony are the main PIs based over in Northern Ireland. And so we have some fantastic clinicians involved as well, and a little bit of help from across the Atlantic from Colorado, from Professor Mark Boyden too. While I'm thanking people, I also need to thank all the people who very kindly funded this work. So I've mentioned bubbles very briefly in the title, and so I wanted to set the scene just by talking about where this technique has come from. So as many of the people listening are probably aware, bubbles have been used in medicine for quite some time now, nearly 40 years, as a contrast agent for imaging. So this story may be slightly apocryphal, but I really like it, and I'm sure there's an element of truth in it. But back in sort of the late 1960s, when ultrasound was a relatively new imaging technique, apparently a scan of the heart was being done with what was then a very fancy new technology, simultaneously with a study of cardiac output using a dye. So a green dye was being injected into the heart. The amount coming out was being measured, and simultaneously an ultrasound scan was being done. And interestingly, it was observed that every time the dye was injected, the ultrasound scan got brighter, the amplitude went up. And so this intrigued everybody, and the dye was patented, and there was a huge amount of excitement over this new contrast agent that had been discovered, until a little bit later it was figured out that it was nothing to do with the dye at all. Actually, what was happening was because the catheter was very, very long, there was a big pressure drop along that, and tiny bubbles were being injected with the dye every time, and it was those bubbles that were actually producing this contrast effect. Now, they weren't particularly impressive scans at the time, but you can see very roughly here that at least there is a quite significant increase in brightness. So, and it was the bubbles that were producing this amazing effect. Now, I need to allay the fears of any scuba divers who are listening. Very crucially, the bubbles I'm talking about are very, very tiny. They are a couple of micrometers in diameter, so they are about a quarter of the size of a red blood cell. So they can safely pass through the circulation without the risk of causing an embolism. So unlike exposing someone to decompression sickness, where you form large bubbles randomly in the body and there is a very large risk of causing a blockage of flow of blood, these bubbles are very, very tiny. Because they are so tiny, they're subject to an absolutely huge surface tension. So to stop that from causing the bubbles to disappear within a fraction of a second, the bubbles are coated. So there is a layer of a surfactant, which is typically a lipid or a protein, sometimes a polymer, that stops the gas from escaping and also reduces that surface tension. Now, the original microbubble contrast agents contained air. Modern contrast agents now contain a much more insoluble gas. So typically it's a perfluorocarbon, has a high molecular weight, and that makes the bubbles much more stable once they're in the circulation. Now, the really exciting thing about bubbles from my perspective is they're not only amazing imaging agents, we can also use them for drug delivery. So we're exploiting that coating on the bubble as a means of encapsulating drugs within each bubble. Now, depending on the chemistry of the drug, we may incorporate that into a layer underneath the shell, or sometimes we can tag it onto the surface, so it depends on the drug. We can also attach things to the bubble surface to enable us to do targeted imaging. So ligands, antibodies can be stuck onto the bubble surface so that the bubbles will accumulate in a particular target region. And so the really nice thing is we can track where the bubbles are flowing through the bloodstream with low intensity ultrasound. And then when they've reached the target site, we see that they've accumulated. We can then increase the ultrasound energy, pop the bubble open, and the drug is released. So we have a way of controlling when and where a drug gets released, and therefore hopefully maximizing the amount that makes it to a target site and reducing the amount that causes side effects elsewhere in the body. Now, the key thing about bubbles, both for imaging and for drug delivery, is how they respond to ultrasound. So as I've said, the bubble itself is really, really tiny. It's a couple of micrometers. This is just showing you how it compares in size to blood cells, so to a white blood cell, a red blood cell, and a platelet. But because it's full of gas, it's incredibly compressible. So when it's hit with an ultrasound pulse, the change in pressure causes the bubble to expand and then contract. And it's that movement of the bubble, it's these oscillations that are responsible both for its the benefit of bubbles as an imaging agent, and as I'll explain in a second, also for drug delivery. In terms of imaging, those oscillations of the bubble cause the bubble to act as a tiny little source of sound. So this is an ultrasound scan, and what you're seeing is the image is getting brighter because the bubbles are washing through into the blood vessels, they're oscillating, they're producing these strong echoes, and we get this really nice contrast between the blood vessels and the surrounding tissue. And actually we can do that now down to very, very fine vessel sizes. With new super resolution techniques, this can even now be down to sort of micro capillaries. And it's those oscillations that we're also using for drug delivery. So what you're looking at here is an ultra high speed movie. So these are taken at a few million frames per second of a bubble that's oscillating in an ultrasound field. This bubble is roughly 2 micrometers in diameter. What you're looking at on the right, though, is what's happening to the fluid around the bubble as it's oscillating. So the bubble is sitting in the center here. We've doped the flow with some fluorescent nanoparticles. And as you can see, as the bubble's moving, it sets the liquid around it into motion. And if you imagine those nanoparticles are drug molecules, you can see that they're being pumped around. And that's really exciting because of the length scales. So although our bubble is only 2 micrometers, the distance over which it's able to transport the drug is 100 times that. And that's incredibly useful because what we can do is we can use these bubbles as effectively little micro pumps that help us to push the drugs out of the blood vessels deep into the tissue. I just want to show you an example of this. This is an experiment that was done by my colleague, Bob Carlisle in Oxford. So this is a section through a tumour. The red is the blood vessel coming through the tumour. Blue are the cancer cells, and the green cells are those that have been successfully treated. So in this case, the treatment was an oncolytic virus. Doesn't really matter, this applies to any sort of drug or particle. And as you can see, we're quite good at treating the cells close to the blood vessel, but the cells that are far into the tumour, we're only treating a very, very small number. And this is one of the reasons why it's very, very difficult to get a curative effect with a lot of cancer drugs. If we do the experiment again though, and now we inject both the drug and microbubbles, and then we expose the tumour to ultrasound, we get a dramatic improvement both in the distance that we can push the drug molecules, so we're going from 50 to 200 microns, and as you can see in the quantity of the cells that we're able to treat. So this micro-pumping effect of the bubbles is a really, really powerful tool for improving delivery. Now, we're very, very fortunate. The building I'm sitting in at the moment is bang smack in the centre of the medical campus in Oxford. So we've got 3 hospitals within an easy walk, and we're very lucky to work with lots of clinicians. And as you can see from this, we're working on a lot of different applications for bubbles. So in cardiovascular, in infection, in orthopedics. What I want to focus on today, though, is cancer, and specifically pancreatic cancer. Now, again, as most people listening are probably very well aware, pancreatic cancer remains one of the most lethal forms of cancer. The 5-year survival is absolutely horrible. We've not really been able to improve treatments for the last 60 years. It's effectively a death sentence. And as a treatment problem, as a drug delivery problem, pancreatic tumours are absolutely horrible. So they tend to be diagnosed very, very late, when they've already grown to a substantial size, which makes surgery very difficult. Poorly perfused, so it's very difficult to get sufficient concentrations of drugs into the tumour. Also, as a result of that poor perfusion, we have very low levels of oxygen, and that tends to mean that those cells that can be reached with drugs are actually more resistant to treatment. And in fact, hypoxic cells tend to be resistant to drugs, to radiation, and even to sort of more modern forms of therapy, such as immunotherapy. Pancreatic tumours in particular have this very, very dense matrix that the cells are embedded in. It's really hard to get drugs to diffuse through them. Often there are secondary tumours, there's metastasis, and sadly, typically patients tend to be quite unwell and unable to tolerate very, very aggressive radiotherapy or chemotherapy regimes. So this is really the reason why we're working with the team in Northern Ireland, is they have a type of drug that has a much, much better toxicity profile compared with conventional chemotherapy. And this is known as so-called sonodynamic therapy. John briefly mentioned in his talk photodynamic therapy. So this is exactly the same idea. You have a drug that can be activated locally. In photodynamic therapy's case, using a laser, in our case, by the application of ultrasound. And this is great because we can focus ultrasound very, very tightly in tissue. We can limit the area over which the drug is toxic. So we can inject the drug systemically, but we only get toxic effects in the target region. There is one snag, however. These drugs work by producing reactive oxygen species. And to do that, we need to have sufficient quantities of molecular oxygen present in the tissue for the drug to work. And obviously, as I've just said, one of the main characteristics of pancreatic tumours, and in fact, a lot of solid tumours, is they tend to be very hypoxic. So our collaboration actually came about at a conference. This was back in 2015. We were chatting after dinner, and John Callan, who leads the group in Ulster, asked, was it possible to put a sonodynamic therapy drug onto a bubble and load it with oxygen? And perhaps because it was quite late in the evening, I rather foolishly said, yeah, no problem, we can do that. Turned out to be significantly more of a problem than I'd realized. There is an extremely good reason why high molecular weight, low solubility gases are used for ultrasound contrast agents. It's because they make very stable bubbles. I knew this. Oxygen, on the other hand, is very soluble in plasma. It's also very small and diffuses very, very quickly. So the engineering of a bubble that would entrap oxygen for a sufficient amount of time to make it through the circulation was not straightforward. And I'm eternally grateful to the fantastic PhD student who did a lot of that work, make it work. Anyway, we were eventually able to do this. So we were able to produce oxygen-loaded bubbles, and we were able to tag these sonodynamic therapy drugs onto the surface. So we did a lot of work to check that these bubbles were enabling us to produce reactive oxygen species when we put it into a hypoxic environment. That was all looking pretty good in vitro. So we then moved into an animal model to see how this would work in vivo. So the model we were using was a human pancreatic tumor in a mouse, relatively simple model. We wanted to give ourselves the absolute best chance of this working in the first study. So we were injecting the bubbles directly into the tumor in this case. That's not, I'll talk about how we get over that a little bit later. And then we were exposing the tumors to ultrasound. For those of you interested, these are the parameters we're using. So these are pretty low energy. This is not high intensity focused ultrasound surgery. These are much, much lower energy levels. And in fact, not significantly different from what you would use for imaging, just rather longer pulses. And these were the results of the first pilot study. So the red line is showing the untreated tumor. So it's a fairly rapidly growing tumor. And the orange line is showing what happens if we inject bubbles and drug, but we don't expose to ultrasound. And as expected, it doesn't do anything. When we do expose to ultrasound though, we're using conventional contrast agent bubbles, so these contain a perfluorocarbon. We do see an effect, the drug is effective, but the tumor starts to regrow pretty rapidly. When we repeat the experiment though, and we use our oxygen-loaded bubbles, we saw a significantly different effect. So the tumor volume reduced by a half, and at least over the course of this quite short experiment, we saw that was retained over 5 days. So for a pilot experiment, that was pretty exciting. And the colleagues we're working with in surgery are very excited because what they would like to use this for first is a means of reducing tumor volume to facilitate surgery. But clearly this was a pilot study. We've since done a lot of work. So we had a few challenges. The fundamentals of sonodynamic therapy, which I'll talk about in a second. Looking at whether there might be some synergies with existing therapies. So as I've mentioned, bubbles allow us to greatly reduce the quantity of drug that we use because they enhance delivery. So we wanted to see if putting a very small amount of a conventional chemotherapy drug in might actually have a synergistic effect. We needed to do a lot of work on our bubbles to make sure we could use them for systemic injection rather than going directly into the tumor. And we also need to look at some more sophisticated tumor models that would give us a better indication of how this might perform in a human. Now, first of all, the issues with the mechanisms. So this only came up when we started trying to publish our initial study. And it was made very clear to us that this was a problem we need to solve extremely quickly. As I mentioned, the drugs are very, very well accepted in the context of photodynamic therapy. We're not doing anything to the compounds. The only thing we're changing is we're using ultrasound rather than light to activate them. The problem was, whilst everybody understood the chemistry of how photodynamic therapy works, it was a complete mystery as to how sonodynamic therapy worked. And as a reviewer was very quick to point out to us, this was the Wikipedia entry relating to sonodynamic therapy at the time. It didn't have a very good reputation, and we were rather horrified by this. I was then very fortunate to have another fantastic PhD student, Estelle Begin, who really made it her mission to sort this problem out and find out why we were getting activation of our drug. And very interestingly, it turns out there was nothing really to figure out. The activation process was exactly the same as photodynamic therapy. The surprising bit was how we were generating the light. Now, the video you're looking at here is a phenomenon that's been known about for a very, very long time, so-called sonoluminescence. And what's happening is that as the bubble is oscillating the ultrasound field, every time the bubble compresses, the gas inside is subjected to a very high temperature and a very high pressure. And those conditions in the core of the bubble can actually be sufficient to generate light production. As I say, that wasn't controversial. That had been known about for a very long time. What was controversial was no one, including me, thought this was possible with the very tiny bubbles that we were using and the sorts of ultrasound frequencies. This is something that's used in chemistry, where you use very low frequencies, big bubbles, and you have to have quite closely controlled conditions. But what Estelle was able to show is that actually microbubbles at medical ultrasound frequencies produce a lot of light over a pretty wide range of wavelengths, and plenty to activate these photodynamic therapy drugs. And yeah, fortunately, we found that the light emissions, the bandwidth of the light emissions was just right to activate these drugs that we were using. So that was great. We were able to satisfy our reviewers that this was legitimate. It's also got quite an interesting potential set of other applications, because the ability to generate light at depth in tissue could be useful in lots of other applications, because a huge advantage of ultrasound compared with lasers is the depth to which we are able to penetrate into tissue non-invasively. But anyway, coming on to that, so we solved the problem of the mechanism. We then went on to look at what happened if we loaded another drug onto the bubble surface. So in this case, we went with gemcitabine. Sorry, excuse me, we did that later. First of all, we tried fluorouracil 5-FU, which is quite widely used as a chemotherapy drug, including in pancreatic cancer. And we did see this synergistic effect. So even though we had very, very tiny quantity of 5-FU on the bubbles, wouldn't do anything systemically, combining it with sondrodynamic therapy, we did see a statistically significant effect on the tumor. What we then found was that when we went to the second drug, gemcitabine, that actually we saw an even more dramatic effect on tumor growth. So this is compared with a systemic dose of gemcitabine, which is what you give in conventional chemotherapy. It slows the tumor growth quite significantly, but it doesn't reduce the volume, whereas combining the two, we see this very, very nice reduction in tumor volume. So this is now something we are taking forward. We've developed a protocol for manufacturing the microbubbles to good manufacturing practice levels. We've got our clinical trial planned, and we're now just in the process of fundraising. Obviously, this year has not been the greatest year to be doing this, but we are keeping going. And hopefully this is something we'll be looking at for a first-in-human trial very, very soon. And say well, obviously, initially, we want to be looking at safety primarily, but also we want to check the quantity of drugs that we can deliver to confirm that we will be able to see the similar tumor response that we've seen in the animal model. So hopefully I'll be able to report back on that in the not too distant future. I just wanted to talk about some of the other things we're doing with this technology as well. So bubble targeting is a big challenge for us. Say one of the big risks is that we will not be able to get sufficient drug into the tumor to be effective. And so we've been looking at incorporating some other things into our bubbles to help that along. I mentioned that we can stick antibodies on the surface. We have a couple of projects involving that. But the other technique we're using is to make our bubbles magnetic. So we actually steal another contrast agent, in this case, an MR contrast agent, and we incorporate iron oxide nanoparticles into the bubble shell. And what this allows us to do, as you've just seen in the video, is by using an external magnetic field, we can manipulate the position of the microbubbles and we can concentrate them at a target site. Now, why are we using this approach as opposed to an antibody targeting approach? Well, the answer is simply efficiency. So this is an experiment where we have a 200 micron capillary, a fake capillary. This is made of cellulose. And we've coated the capillary with avidin, and our bubbles are carrying biotin. So we have a biochemical targeting mechanism. And we want to see how many bubbles get stuck to the side of the tube. So as you see, as the bubbles start to flow in, we see plenty of them starting to stick to the side of the tube, which is looking pretty good. If you look very, very closely, you'll see a few of them get knocked off by the flow. But if we do the experiment now using magnetic bubbles, and we've got a magnet sitting up here. So we're running the experiment over the same timescale. Looks pretty similar initially, but over sort of 10 to 15 seconds or so, we're able to accumulate a much, much larger quantity of bubbles with the magnetic technique. I mean, as you can tell from the image, many orders of magnitude more bubbles, which hopefully translates into a much, much higher order magnitude of drug. So that's why we're pursuing this. They're not mutually exclusive. We can make bubbles that are both magnetic and antibody targeted. So we've gone back and we've done an experiment, again, trying to deliver sonodynamic therapy in a mouse model. This time we're injecting the bubbles systemically, and we're using an orthotopic tumor. So this is a tumor that's actually in the pancreas of the mouse. So at least in theory, it should be a more representative tumor model. I'm very happy to discuss the challenges associated with what constitutes a representative tumor in a mouse. So that's a big problem, but for this experiment, it was fine. So we inject the bubbles. We're then applying a magnetic field above the tumor. I'll say a bit about how we do that in a second. And then we're exposing to ultrasound. So same ultrasound conditions, but we're now injecting systemically and using magnetic targeting. And we're looking, in this case, over a much longer time period compared to our pilot experiment. And what we see is that in the targeted case, we are able to see a sustained reduction in tumor volume. And this is from a single treatment. And actually, the sonodynamic therapy alone in this case didn't give us a sustained effect. Now, tumor volume doesn't tell the whole story. As I mentioned, pancreatic tumors in particular have a very dense extracellular matrix. They're packed full of basically dead tissue. So we also extract the cells to look for apoptosis. So active caspase is a marker of cell death within the tumor. And what was really exciting here is we saw that was a very, very significant change. It looks like the drug's been extremely effective in the targeted case. Now, moving on to the really big question. Magnetic targeting is great. As an engineer, it's very exciting to me. There's one huge problem, and that is the force that you get from a magnet drops off with a cube of distance. So unfortunately, showing that this works in the sort of millimeter centimeter scale in a mouse doesn't tell us a huge amount about what's going to happen in a human. So one of the things we've been working very hard is to try and understand, can this ever work on the many centimeter scale? So this is a very basic setup that we had in the lab, and I had another poor student who spent many months playing with this. I do need to say something about the magnet. So we can't just use a simple magnet. All that would do is it would suck the bubbles towards the skin, and for significant tissue depths, that's just not going to be remotely successful. So what we have instead is actually an array of permanent magnets. So these are very, very powerful neodymium iron boron magnets. So if you've ever played with them, they sell lots of executive toys producing this material. They are astonishingly dangerous if you stick them to the wrong thing. We have them set up in an array such that the orientations of the magnet varies across the array. And what this allows us to do is combine sort of push and pull forces so we can confine the bubbles at a particular point. And we do that so that we can get the bubbles located at the right depth in the tissue. So in this case, our tissue was just a block of gel. We had a tube running through it, and we were pumping bubbles through the tube. We're imaging them with ultrasound, and then looking how many bubbles we were able to hold above the magnet. So we kept changing the flow rate, pulling the magnet further and further away to work out what are the limits for targeting using a magnet. So these are the images that we see on the ultrasound scanner. So the bubbles start washing in, or hopefully they do, if the video will work. Yeah, there we go. Be patient, Eleanor. There we go. So you see the bubbles washing in, they're being deflected down, and then we're seeing this nice bright up here, which means we've been able to track bubbles along the base of the tube. If we take the magnet away, they all float away again. Fortunately, what my student Josh was able to show was we were able to get trapping at sort of distances of 5 or 6 centimeters, which is plenty for the applications we're interested in, and actually at really quite high flow rates. So certainly more than you'd ever expect to get in the capillary network around a tumour. So that was great news. That looked very, very encouraging for us going forwards to apply this to humans. But of course, a block of gel is very, very far from real tissue. So we've been collaborating with another team in Oxford. So my colleague, Professor Konstantin Kousios, and Professor Peter Friend in the Department of Surgery have developed this amazing system for maintaining a transplant organ over up to 72 hours. Sorry, this slide I said 24 hours, then corrected, it's 72 hours. So the organs can be kept preserved to enable transplants. But what it allows us to do is to try out our drug delivery technologies in a system which is really as close as you can get to a real organ. In this case, it was a pig liver, but we can do this with discarded human livers as well. And we repeated the experiment. So we were injecting our bubbles into a blood vessel. We had a magnet positioned underneath, and then we're using ultrasound imaging to see whether we get that retention technique. And also, crucially, whether we also saw movement of the bubbles into the tissue that we wouldn't want. So this is what we saw. So again, we're looking at the bubbles washing into the vessel. And to our intense relief, we saw this nice white bright up along the lower surface of the vessel. This is the reflection from the magnet that you're looking at down here. We got our orientation a little bit wrong. We were actually about 4 or 5 centimeters away from the vessel. We thought we'd intended to be about 2 centimeters, so we were very lucky this did work. But we were able to confirm that we can see this in real tissue. So again, this is something we're sort of hoping to take forward. Obviously, there are some safety concerns associated with the use of nanoparticles that we need to be very confident on. So some of the other things that we're looking at is efficacy in different types of tumour. So some of the urologists we're working with are very, very interested in this as a means of treating prostate cancer, so avoiding sort of having to have radical prostatectomies. And we see a very similar response, which isn't a huge surprise because we're producing reactive oxygen species. We don't expect this to be particularly tumour specific. In this case, we were combining it with a different drug, with paclitaxel. But yeah, we see this very nice reduction in tumour volume. In fact, in this case, a really, really significant change. Now, one of the last things I wanted to share are some newer results. So these are a little bit preliminary, but they are very, very exciting. On my challenge slide for pancreatic cancer, one thing I didn't mention or haven't addressed yet is metastasis. So the spreading of the primary tumour to other sites. And again, I'm sure most people listening are aware, it's those secondary tumours that are often fatal. Now, we can't necessarily stick bubbles to all of the secondary sites because often they're too small to actually be detected. But what is very, very exciting is sonodynamic therapy appears to produce an immune response. So it seems to actually be able to impact other tumours remotely through an immune pathway. We have yet to establish which one. So these are some results from an experiment where the animal has 2 tumours, one on each hind limb. Bubbles are injected systemically, but only one tumour is subject to ultrasound. So the green curve here is showing the tumour that's actually treated. As we'd expect, we see an effect of the sonodynamic therapy. The red tumour is the one that has not been exposed to ultrasound. And what's really exciting is that seems to be showing quite a significant response as well to the control, to the untreated case. And what John and Tony in Ulster have done recently is they've combined that now with an immunotherapy drug, anti-PD-L1, seeing that there is a synergistic effect. So in this case, all these curves are for the tumour that's not exposed to ultrasound, but they're all showing a significant effect compared to the control. And again, that's something which we are very, very actively exploring and hope we'll be able to combine into our later clinical trials. So just to sum up, we're very excited about the potential of sonodynamic therapy in combination with bubbles as a targeted cancer therapy. We've shown that by engineering bubbles to carry oxygen, this seems to significantly enhance its efficacy, and that we see this synergistic effect with very low doses of conventional chemotherapy as well. say excitingly also the potential for an immunotherapeutic effect too. By doing some more bubble engineering, combining magnetic particles into our bubbles, we're able to achieve some additional targeting effects. Hopefully that's going to be something we can wrap into a new treatment for pancreatic cancer going forwards. So with that, thank you very, very much indeed. I realize I think I'm the last of the day, so people are probably looking forward to a nice cup of tea, but I'd be delighted to take any questions or discuss any of the points raised. Thank you.
Awesome.
Thank you so much.
Really good. Laura's going to lead the questions. So Laura.
Yeah, thank you so much, Professor Stride. I agree with Neil. That was very good, very informative talk. I think especially as a medic as well, I can definitely sort of find that very useful. If any of our attendees have any questions, please do type them into the chat. In the meantime, I have a question for you, if that's right. So I was wondering, what type of time scale do you expect for sonodynamic therapy to be available routinely for cancer treatment in the UK within the NHS?
That's a really good question. So the interesting thing about it is because it is an existing drug, this is already approved. There are lots of photodynamic therapy drugs approved, possibly quite soon. And actually, there's a group in Canada, in Toronto, who've picked this up really quickly. So we know them very well. They do a lot of very beautiful ultrasound therapy studies. I mean, I think they're planning on trialing this in brain cancer very, very soon. So it's an interesting one because the drug's already been through all of the safety and the approval, both you know, in the UK, in the US, and around the world. And ultrasound therapy machines are also approved. We're hoping that's going to lead to a relatively smooth translational pathway. Yeah.
Brilliant. So would you say in terms of a few years or maybe 10, 20 years? What
I would, well, I sincerely hope less. So if funding conditional, I mean, we're hoping to run our first study next year. If that's successful and everything is shown to be safe with this combination, you know, we'd be hopefully looking at more like 5 years.
Oh wow, okay, that sounds brilliant then.
Fingers crossed, but that's our aim.
And you mentioned pancreatic cancer and prostate cancer as well. Are there any other specific types of cancer that you think this therapy would be well suited to?
Yes, so I say in principle, this could be applied very broadly. I know we've tried it in breast cancer actually already, and that showed very, very similar results. Very interested to try it in brain cancer. So one of our other projects is on treating brain metastases. And again, toxicity is a big, big issue there. This would be very exciting if that's shown. I think the only cancer for which this is less suitable is lung cancer, simply because ultrasounding the lungs is quite dangerous. Having said that, if you do have quite an advanced tumour, that region of the lung may be quite fluid filled, at which point you can apply ultrasound to it quite safely. But I think lung cancer is probably quite a long way down the line, but most other solid tumours, definitely.
Okay, brilliant. And just one more thing. So this kind of route of drug administration, would this be ever considered, do you think, to treat other conditions other than tumors, solid tumors?
Good question. So we have a project looking at it in the context of antimicrobial. So it's an alternative to antibiotics. So we're looking at that, obviously looking very carefully at its potential resistance as well. Again, the reason we're interested is because it works through reactive oxygen species generation, resistance should be less than you would get with conventional antibiotics, but that's certainly an area we want to look at. There may well be others. I mean, photodynamic therapy has been applied to a lot of other conditions. So yeah,
Oh, brilliant. Okay, that sounds great. And a very promising sort of new type of therapy. That's not, yeah, hopefully. That's a very good talk. Thank you so much. And we've been getting a lot of positive feedback on the chat as well for the talk. So again, thank you so much. That was very informative.
I was wondering if I could sneak in a question. So I'm a gastroenterologist by trade, almost done with my training, and it never ends. I was just thinking actually the applications of what you presented there. So say pancreatic cancer, completely agree, big issue, poor morbidity, mortality. And the focus as say a physician are the pancreatic cysts. So kind of the cysts that are pre-malignant, say your IPMNs, for example. I do very basic endoscopies, upper and lower endoscopy. I'm not brave enough to do endoscopic ultrasound, but I was actually thinking, other applications for those that are trained in EUS when they see a pancreatic cyst, as opposed to, say, taking just a simple aspiration and then saying, okay, this is something to worry about, that they could actually provide drug-based therapy at that point, so to prevent it progressing to the cancer.
Absolutely. I mean, that's one of the huge advantages for pancreatic cancer is because endoscopic ultrasound is already very well accepted. We have a ready-made means of applying ultrasound to those. And it would be very interesting to see if, as you say, pre-malignant masses could be treated with this as well, even without the drug. So if you turn the volume up a bit, if you like, and cause the bubbles to oscillate harder, they actually have an eroding effect on tissue. So there's a technique known as histotripsy, which there is some very exciting work going on in Michigan, where they're using this as an alternative to surgery. So as a means of just removing masses, things like uterine fibroids and some other sort of benign conditions. It's a really nice way of doing that. So yeah, it would be great to look at that.
Yeah, no, for sure. Laura, any final thoughts?
That was brilliant as well. Thank you, Neil, for your question as well. I think it's good to have some quite specific sort of questions in medicine as well when it comes to these things. But yeah, I don't think there's any more questions. So once again, thank you so much on behalf of the Clinical Engineer Hub. That was a very good lecture, and thank you as well to Prof. Girkin if he's still on the call as well. Okay, well,
thank you so much for organising. I think we need more of this. It's great.
Yeah, we're very excited. The first 2 days have been brilliant. And
no, absolutely, I've been teaching solidly, so I've been dipping in and out, but everything I've seen has been absolutely fascinating. I need to drop John an email. I've got so many questions.
I'm sure. Okay, so we're just going to close, everyone. So tomorrow is our final day of the Clinician Engineer Hub. We have experts all over the world coming tomorrow as well, across academia and industry. And we also have the World Health Organization joining us. So it's going to make for a very interesting day. So thanks for everyone to join, and we'll see you tomorrow. All the best.
Thank you.
Thank you.